for having me. I'm excited to tell you about some work that we've been doing over the last 7 or 8 years on some redox active nucleic acids and how nucleic acids can behave in some hypersaline environments, kind of fitting with the ROS flavor of the meeting. So just to kind of frame the talk, this is from a paper Jerry Joyce published about 20 years ago, sort of positing a timeline for the emergence of life on Earth. And that's kind of that underlying part, which is where we think this prebiotic chemistry area kind of going into an RNA world. I guess I would preface that with, kind of thinking about what was said in some of the other talks. I'm not as hardline about, this necessarily being a pure RNA world. I think that there probably was co-emergence of biopolymers, and these were probably interacting with one another. How they might interact, how they might fit in different environments is some of what we think about here, and I'll talk a little bit about in the talk here. We're mostly thinking about RNA, but I think that there's parsimony would dictate, I think, evidence in history, the ribosome, the spliceosome, different machinery that, has a lot of RNA in it, that they probably were coexisting. So a lot of this is going to relate to Martian scenarios for the origin of life. I think some of this kind of ties back to Earth, but just sort of motivating that aspect of it and some of the hypersaline story. This goes back to 2008 when this was the first discovery of a species on Mars called perchlorate, which is broadly a member of these compounds called oxychlorine ions. So what happened here was the group that did the work went up and they were looking for nitrate in Martian soil or regolith. They got a result that didn't make any sense. It looked like there was actually more nitrate in the regolith than there was the mass of soil they scooped up, which of course didn't make any sense. And with some really clever experiments, they sorted out that there probably was oxychlorine species, in particular, a lot of perchlorate on the surface of Mars. And this has since been confirmed by several other missions at different sites on Mars. It's now pretty widely accepted that there's a global distribution of oxychlorine compounds, especially perchlorate on Mars, at a concentration that's pretty high. It's about 1/2 weight% in the regolith. So what are the consequences of this? Perchlorate it's a weird compound. This is really surprising because, it's toxic to a lot of living things, which isn't great. There are some microbes that can breathe perchlorate. They can use it as an electron acceptor, just like we breathe using oxygen. One of the really cool things about it is that it does stuff like this. So if you're a bench scientist, there's a good chance you've encountered this. This is NaOH, so not perchlorate. It does the same thing, though. I'm showing this because it's probably the example that you've seen, and that's that it does this thing called deliquescence, which is kind of just a $10 word for saying that if you take one of these salts, NaOH here, perchlorate does it as well, leave it out on the bench or in an atmosphere with an appreciable amount of moisture, it'll soak it up out of the atmosphere. Perchlorates do this too. If you leave a perchlorate salt on the bench, you'll end up with a puddle afterwards. And what that puddle is, of course, is the salt you started with, plus water that's it's basically soaked up from the atmosphere. This could provide a source of liquid water. Flip side of it, of course, is that the liquid water is going to be really salty. And so what does this mean for life? Could this be, a refuge for life? Could this be something that life exists in? There's people working on that with, known forms of life, or there's salty forms of life. Dirk Schultz has done some nice work showing that there's some fungi that can exist at surprisingly high perchlorate concentrations. What we were interested in is what's the impact of this on molecular evolution? So what happens if we just take some biopolymers and put them in hyperchlorate? There's reason to think that there might be a downside to this. This is a ranking of ions called the Hofmeister series. You don't have to follow every single one here. You can just see that generally ions follow kind of this ranking here where you go from kind of the green end where they're called cosmotropes. They tend to stabilize protein and nucleic acid folding over to the red end called chaotropes where they destabilize it. You can see that they're kind of exemplified by on the far right there, perchlorate. Franz Hofmeister was the discoverer of this. You can see that there's actually a plaque commemorating the Hofmeister series and Franz Hofmeister in Prague. This is showing chlorate ClO3. It's kind of the far right there. These are known to be really destabilizing compounds, nitrate being among them as well. If you look at biopolymers in these Hofmeister ions, you can see that they kind of follow this stabilizing and destabilizing trend here. So this is looking at the melting temperature of sort of a model protein that's used in a lot of these systems called lysozyme. What lysozyme is just a protein that has a pretty well-behaved unfolding transition. Not being used so much for the function here as something that's just, it's really straightforward to observe how it behaves in the presence in different salts and solution conditions. And what you can see is that we're looking at that middle line there, which is basically as you add the salt, the melting temperature doesn't change. So going from no to 1 molar chloride, it's relatively, inert. It's not stabilizing. It's not destabilizing. If the line goes above that, that means it's stabilizing. If it goes below it, that means it's destabilizing. And so you can see going above it are things like sulfate and phosphate. Those would be things that were in the green side of that trend line there, that trend that I showed you. And then on the right were the red ones, things like iodine and perchlorate. You can see as you add those in, the more you get there, the less stable the protein fold is. So this is kind of just got us thinking about what might this mean for life in a really saline environment. There's been a lot of work on how did all that perchlorate get on Mars there? And the current thinking is if you have a dry oxidizing world for a long time, you probably will get a bunch of perchlorate there. We don't have a ton of perchlorate on Earth. There's a few places that have appreciable amounts of it, the Atacama Desert being one of them. But we do have organisms that have evolved to handle a lot of salt. And just kind of looking at what they've done, there are some that sort of clean up their house by ejecting the salt, exemplified by the kind of upper left there. They have ion transporters that sort of pump salt out. Then there's ones like in the lower right that deal with having kind of a salty cytoplasm. You can see I have it shown as their biopolymers are salt adapted. What that means is that a lot of the proteins in those organisms, in order to become salt adapted, they have a lot of charged residues on their surface. That makes them strongly hydrated. That means that they can get along with salt pretty well. Most of the work here has been done with protein. A lot of the work that we've done here is with RNA. I'll talk about in a minute here. One other interesting aspect of this I want to touch on first, though, is one of adsorption. And that's that if you take a lot of these chaotropic salts, one thing that they can do is cause nucleic acids to actually adsorb, basically stick to anything silica-based. So glass beads is kind of the archetypal example. This is something that we use in labs for purifying DNA. If you take some of these chaotropic salts and silica and DNA, it'll stick to the silica. You can wash it with ethanol, it'll stay stuck to it. When you go to low salt conditions, it'll come off. And this is a technique that's used in molecular biology labs all over the world for purification of plasmids, PCR products, things like that. I mentioned, silicate or silicates generally do this. Glass will do it. People have done it with crushed up glass. Basalt, kind of lava rock is one example. This is sort of a good model, it's thought, for Martian regolith. So one thing that we've done is looked at, nucleic acids will actually stick to sort of simulants of Martian regolith. So things that are made out of basaltic glass. The mineralogy of this Martian regolith is such that there is a lot of silica. You can see it's about half weight percent silica. And what we found is that this is basically showing, that nucleic acids will stick to both the Martian simulant as well as silica. So this is basically looking at a gel showing that the SM there is showing what we're starting with. The 1 and 2 are those ethanol washes. You don't want to see anything there if it's actually sticking. And 3 is that water elution. So basically what you would be doing with one of these plasmid mini preps, you're basically getting the DNA back. The MGS1, the right 3 lanes there are that Martian regolith simulant. That's showing that this actually acts a lot like this. This is something that, we're thinking about what this could mean, because if you have a really saline environment, this is something that could impair things like compartmentalization, because it can cause problems with lipid self-assembly. But this is kind of a way of immobilizing things. If you have things stuck to glass, you sort of reduce this dimensionality problem to something closer to 2D because you've kind of limited diffusion there. So that's one interesting aspect of it. Another aspect that we thought a lot about here is sort of what are the consequences for nucleic acid folding? We're interested in, things that RNA can do. Thinking about examples of ribozymes that exist in extant life, things like the hammerhead ribosome, the ribosome, the spliceosome, or the group 1 intron. There's examples of catalytic RNA. What might happen in these really salty environments? And so just looking at nucleic acid folding with a lot of perchlorate around, what we found is, broadly like what I showed for the lysozyme example, you can see that as you add in some duplex DNA and take an increase in concentration of perchlorate, you can see that broadly you're seeing a decrease in that melting temperature. So that line going down means that the duplex is less stable. You can see that there's an initial increase there, and this is kind of consistent with what you expect to see with nucleic acids and salts. Salts generally stabilize nucleic acid duplexes, but once you get past about 1 molar or so, you can see that the stability kind of starts to go down. One thing that's interesting is we found that this actually isn't true of all secondary structures. If we take another special nucleic acid structure that can form a 4-stranded structure called the G quadruplex, you can see that it's broadly kind of the same trend, but it's a lot more muted in terms of the impact of some of these perchlorate denaturants. You can see that you still see that kind of increase with a little bit of increasing salt. As you go to higher perchlorate, it starts to go down, but not nearly as much as the duplex there. And so, perhaps not surprisingly, this is consistent with what you see with something where you have sort of a different folding element for each sort of secondary structure. In a duplex, you've got base pairs. In this quadruplex, you have stacks of 4 purines. So this is really about double the size or a little bit more of Watson-Crick base pair. And one consequence of that appears to be the fact that you actually get kind of different folding rules in high salt than you do in the lower salt. What we found is that this can actually let us kind of switch back and forth between different secondary structure states. So this is kind of showing a cartoon of a DNA system where that red strand is really G-rich. It can fold into this 4-stranded structure, but it also can form a duplex when it's in the presence of its C-rich complement. So that would be that blue strand there. And so what we've set up is kind of a fluorescence monitoring system where we can look at it in the duplex state, in that 4-stranded state, or in a single-stranded state. And what we find is that depending on the salt concentration, we actually get some really complex behavior in terms of which folding state you can get. So we're starting with the red trace there. We're going from, a state A there on the right, which is the double-stranded DNA, a state B, which is that 4-stranded structure, and then a state C, which is where everything is unfolded. And you can see that in that, lowish salt, the 1 molar, we start with a duplex. It's briefly in that G quadruplex state as we heat it up, and then everything unfolds. When you go to kind of intermediate salts, you can see that you've sort of shifted the preferred folding state, where for most of that liquid range of water, it's actually in that G quadruplex state, and it's only for a fairly narrow range in that duplex state for unfolding into single strand. And then if you go high enough, you'll actually force it only into the state B and C. And so this really shows that, even for something as simple as kind of this toy nucleic acid system, we really can change the folding rules for nucleic acid as we go into some of these different salt conditions. And just kind of thinking broadly about what this means for biopolymers, if we think about something like an evolutionary funnel for them, the going through different, salinity states could have a pretty powerful impact on molecular evolution. Another thing that we've looked at is, okay, we have a system where we can monitor folding. What can we do in terms of sort of functional behaviors? One thing that these 4-stranded structures can do is that they can bind to heme, so the same cofactor you find in things like hemoglobin and horseradish peroxidase. And that complex between the 4-stranded nucleic acid structure and heme can actually act as a peroxidase or an electron transfer catalyst. Sort of exemplified by this reaction that we have with a dye called Amplex Red that goes from colorless to a red fluorescent dye when it reacts with one of these. One thing that we can see is that in that kind of intermediate salt state, that 5 molar salt, we see that we actually can get that redox active complex where we can get some of this structure switching going on here. And so this is another case where depending on the salt condition, we can actually tune, actually functional behavior of some of these nucleic acids, including doing some interesting redox or ROS related chemistry. We've also found that surprisingly, one thing that comes up when I talk with a lot of people about this is, perchlorate, isn't that something that's so strongly oxidizing? Isn't it going to chew everything up? What we found is that perchlorate is surprisingly metastable. That's to say that even though it is a highly oxidizing molecule, it's kinetically stable to the point where we can take these molecules and heat them up, as I've shown, basically recover them. They retain function, and looking at conditions where we can probe how oxidizing they are, they're not really appreciably more oxidizing than just the aerial oxygen that's around when we do some of these experiments. Another thing that we're really excited about is just kind of looking at some other chemistry that we can do with this. One thing that we've done here is, most of the work that we've done is with perchlorate, but if you're noticing at the beginning, I was kind of careful to say oxychlorine species. There's a whole suite of compounds going from chloride, Cl minus, to the -1 state, up to perchlorate, which is chlorine +7, or the most oxidized state you can get chlorine into. You can have most oxidation states in between there. You can have hypochlorite, you can have chlorate, you can have chlorate. We looked at one of the more active or kinetically active oxidizing species, chlorate, and just looked at, okay, so if we take this peroxidase complex, can we do, maybe some more interesting chemistry with this? Can we do something other than just an ROS-based reaction? Can we do a more complex reaction like a chlorination reaction? And it turns out that we can with at least a couple substrates. This is looking at basically the chlorination of a dye called phenol red. There's kind of a characteristic change in the visible absorbance spectrum when it gets chlorinated. You can see that the pink line there is the RGQ, that's the RNA heme catalyst. Looking at that, you can see that you're getting chlorination that's pretty comparable to HRP, as horseradish peroxidase, a protein enzyme that's known to be able to do this. We can look at this by UV-Vis. We've also looked at it by mass spec on the right. And you can see that looking at it by mass spectrometry, we get multiple chlorination events with the nucleic acid heme complex there. And so this is something we think is really cool, because if we look at nucleic acid catalysts that we have, there's been a lot of work evolving some really sophisticated catalysts, some of which we've used, and I'll show you here in some of the last few slides. But for the most part, what these are doing is phosphate transfer reactions, which are certainly important to biology. If you think about RNA polymerization, DNA polymerization, kinases installing phosphate groups, moving phosphates around, the phosphate economy is certainly important to life. But it's obviously not the only thing. And we think that this carbon-chlorine bond formation is such a linchpin in organic synthesis that this is something that could enable some really interesting chemistry. And this is something we continue to explore and are really excited about in the future. We've also looked at a range of other functional nucleic acids in some of these perchlorate systems, just to see, do these still work? Can we actually get function in perchlorate? And we found that a number of ribozymes do. This is the hammerhead ribozyme. So this is a nuclease ribozyme that undergoes a self-cleavage reaction. So basically what we're looking at here is there's 2 strands of RNA. There's that yellow label FAM, that's a fluorescent label we can use to monitor the cleavage on a gel shift assay. It's going to cleave where that red arrow is. And so these are kinetic plots looking at the cleavage rate under different conditions. And what you can see is that in multiple molar concentrations of perchlorate, you're getting appreciable amounts of cleavage here. So this actually will continue to cleave up to quite high concentrations of salt. We've also done some work looking at this same ribozyme and seeing if we can get some interesting functional behaviors out of it. A few years ago, we did some work looking at sort of some of the functional consequences of having short replication intermediates around that you would expect to be produced, in the time when we weren't very good at doing RNA polymerization. So you can imagine if you're getting random pieces of RNA or maybe copying intermediates, you're going to produce, some of the RNA that you want. So something like that hammerhead ribozyme or an RNA-based RNA polymerase. But in addition to that, because these are low efficiency copying reactions, you're also probably going to get a lot of short pieces out of this. And what we were looking at here was basically, what's the consequence of having some of those short pieces around, and what happens when you have some really simple physical processes? And so what the left is showing there is basically one of these model ribozymes that we're looking at. So the hammerhead ribozyme is what's inside those liposomes in kind of the bluish black there. And the red there are short pieces of RNA. We've done this with ones that are complementary to the RNA, so things that you would expect from like a replication intermediate or even just totally random sequences. And what happens is if you have a lot of random sequence or short RNA around, so sequence related or random, at large excess, so like you would expect to be produced in low efficiency copying reactions, what you end up with is a low efficiency ribozyme reaction. It doesn't work to a large extent because those tend to inhibit the reaction. If they get diluted, it'll actually switch on and you'll get a significant increase in the kinetics of the reaction. This can happen with liposome growth. One thing that we found is that perchlorate can actually enable this too. This is looking at sort of that same system where we have some of those random sequence RNAs. What they'll do is actually inhibit the reaction of the ribozyme. But if we add in perchlorate, that actually will act as kind of a mild denaturant where it'll knock off those short pieces that kind of act as inhibitors, but we actually will get switching on to the hammerhead ribozyme. And so this is something where it actually kind of turns it into a positive. Going to the higher salt actually removes some of those inhibitory compounds that keep a ribozyme from working. We've also found a range of other functional RNAs work in this system. This is a fluorescent aptamer called broccoli, and you can see that up to several molar broccoli, we still get fluorescence out of it. So, not just limited to ribozymes, we can see that an aptamer works. I won't show you today, but we've done some work with the RNA-based RNA polymerase. Those will continue to work in multiple molar concentrations of perchlorate as well. And so another thing that we did here was kind of compare this to protein-based enzymes. What's the consequence of hyperchlorate for protein-based enzymes? And what we found is that it's kind of a mixed bag depending on what you start with. The top there is showing basically kinetic plots for kind of an archetypal protein enzyme. This is EcoR1 or restriction enzyme from E. coli, which is an organism that's evolved to deal with not a whole lot of salts. And these kinetic plots, more slope is working better. And you can see that without having perchlorate around, it worked pretty well. As you add in perchlorate, it doesn't take a lot to shut it off. At 100 millimolar perchlorate versus no perchlorate, you've gone to about, a quarter of the activity. Going to twice that, just 200 millimolar, the enzymes basically shut off. If we look at enzymes that come from some organisms that have adapted to deal with more salt, it starts to look better. The enzyme on the bottom there is a cartoon of a beta-lactamase, or an antibiotic-degrading enzyme from a halophilic bacterium. And what we can see is as we go to high salt with that, that'll hang on for quite a bit longer. It's only at about 2.5, 3 molar that really it kind of starts to shut down there. And so this starts to become comparable to some of the RNAs that we've seen here. What we think is really interesting about this, though, is that if you look at the RNAs that we've looked at, that broccoli enzyme or that broccoli aptamer was selected in E. coli, the hammerhead ribozyme comes from a viroid that inflects avocado. None of these really evolved in high salt. So it seems like there's something intrinsic to RNA that's really tolerating some of these high salt conditions. Going back to the beginning of the talk when I was talking about some of these salt adapted organisms, there are ones that take the salt out, and then there are ones that kind of learn to live with it. Those ones that learn to live with it are doing so in part by putting more hydrophilic groups or charge groups on their surface, which is true, of course, of RNA at every single residue. And we think that this could be really important to salt-adapted biopolymers. Finally, the last thing that we looked at here is renaturation of some RNAs after they've gone to too high a salt. So, all these have some salt condition where they shut off. One thing that we found is that some enzymes will recover from it better than others. This is going from, on the left there, low salt for broccoli, that fluorescent aptamer, EcoR1, that restriction enzyme. Habla is that beta-lactamase that I talked about from the halophilic organism, the antibiotic-degrading enzyme. And then finally on the right there is one I haven't talked about, which is a restriction enzyme from TAX, so a thermophilic enzyme. And what you can see is that of what we looked at, everything will shut off at high enough salt, but when you go back to low salt, they recover a little bit differently. Broccoli, you get most of the activity back. EcoR1 looks like irreversibly unfolded. The salt-adapted beta-lactamase comes back, but not as well as the RNA. And then that thermophilic one comes back entirely. And so it looks like depending on the evolutionary path or the evolutionary state of a protein, they can have different tolerance for these salt excursions. But RNAs generally seem to do pretty well here. So, yeah, that's some of what we've been looking at here with these nucleic acids, both in terms of how they react with different salts, particularly motivated by, a really oxidizing world, as well as some of the behavior that they can do in terms of doing some of this redox chemistry, both in terms of, kind of canonical peroxidase type chemistry, as well as, another formal oxidation, a chlorination reaction, which is, new to ribosomology and something that we're really excited about too, because it seems like this is something that could enable, I think, a lot of things that would be really cool to be able to do in an RNA world. So, Tanner in the center there did most of the work here. Ben, Nate, and Matt also contributed to the project. It's in part of collaboration with the KRISHNENDU lab at the University of Minnesota. I want to acknowledge funding, particularly for NASA and some other agencies. NoRCEL again for inviting me, and I'll
By
use
me, Alan HIDALGO ARANDA, and with the guidance of my master supervisor, Dr. Alicia Negron Mendoza. And we are both part from National Autonomous University of Mexico, and its main campus is University City. And in University City, there's the Nuclear Science Institute, where it is our laboratory of chemical evolution. So a quick resume of what we did is we studied the stability of Uracil in extreme conditions like primitive Earth and meteorites using quantum chemistry, more specifically density functional theory. So this talk will be about first a quick overview of our line of research, that is chemical evolution, and astrochemistry, computational chemistry, and finally how do we use all of these tools to study the stability of uracil using density functional theory. So let's start with chemical evolution. Chemical evolution is in the bottom-up approach. This means that we study from molecules to the origin of life. Uh, Melvin Calvin defined chemical evolution as the process that converts simple organic and inorganic compounds into biochemical important molecules under the conditions of prebiotic Earth. And this is an old definition from the 50s. We now scientists ask more questions about chemical evolution. Chemical evolution could only happen in prebiotic Earth. We know that that's not true. What about other planets, meteorites, interstellar medium? So, how do we study chemical evolution? I think one of the best examples to show how we study chemical evolution is first with the operant Haldane theory that says that biochemical important compounds could be synthesized with biogenic molecules using cosmic rays and UV radiation from the sun as the energy source. They set the conditions of how could chemical evolution could occur. And Urey Miller later in the 50s simulated primitive Earth conditions and managed to synthesize amino acids. And that's how commonly we make our experiments. We set the conditions of a planet, or prebiotic Earth, or meteorites, and then we make some simulations to study those environments. So now I'm going to talk about astrochemistry. Our astrochemistry is the most simple definition, and the one I like the most is chemistry in space. Astrochemistry observes spectroscopic emissions of molecules, and they tries to give the correct interpretation with theories. But that's one of the ways to study astrochemistry. Another way to study astrochemistry is to ask what molecules are in space and how do they react to other molecules and electromagnetic radiation. Astrochemistry is often approached as a three-pillar approach. The three pillars are observation, that's astronomy and spectroscopy, modeling, quantum chemistry and experimental chemistry. This work is in quantum chemistry, and generation of reference data. All of these spectrums that we get from space or spectrums that we get from quantum chemistry, we put them together to then study later and find the molecules that are in space. And the tool that we use in quantum chemistry is computational chemistry. What the main objective of computational chemistry is to describe the properties of molecules using theoretical methods. The main methods that we use are Hartree-Fock is the historical method, but there is Muller-Plissett theory and density functional theory. Those are the one of the most used because they are very in equilibrium with cost and precision. And finally, there are the couple cluster theory methods. Those are highly costly. It takes a lot of time to make those calculations, but also have very precise results. So what do these methods try to do? From the simples, they try to solve the Schrödinger equation. The most simple makes a lot of approximations and it has lower precision, and the more complex it gets the method, it better solves the Schrödinger equation. So how do we use computational chemistry to study astrochemistry and chemical evolution? The first one will be the spectroscopy. In the spectroscopy, there's the bottom-up approximation to the molecules with quantum chemistry. That means that we get to the spectrum using the molecule. Commonly in laboratories, we get a spectrum and try to link it to the molecule. And the other way, that's the way that we use in this work, is prediction and explanation of reaction pathways and mechanisms. We follow from reactants to transition state to products the reactions in outer space and prebiotic air, meteorites, any kind of environment that is out of our reach. So we studied with computational chemistry, density functional theory, the stability views of Uracil. What kind of conditions? The first will be in prebiotic air conditions. Uracil is one of the 5 nitrogen bases that form RNA and DNA. And so that says the conditions, the first conditions, prebiotic air. And also in 2020 to 2023, in the Hayabusa, with the Hayabusa spacecraft, it got some samples of Ryugu asteroid, and from experiments they found that Uracil was in Ryugu asteroid. So how do we study it using computational chemistry? We studied with studying the reaction between uracil and hydroxyl radicals. That's a reactive oxygen species. Why we studied with hydroxyl radical? Because when in prebiotic air conditions and asteroid conditions, it's heavily irradiated. There's a lot of radiation. So when water gets irradiated, one of the main products is hydroxyl radical. So we studied the reactions between hydroxyl radical and uracil. There are mainly 3 kinds of reactions. The one that I'm not going to talk about today is electron transfer reactions. And the ones that we study are radical adduct formation and hydrogen extraction. And I want you to look first mainly in the hydrogen extraction that forms the amidyl in nitrogen 3 and the amidyl in nitrogen 1. Why they are important? Because if uracil gets hydrogen abstracted in nitrogen 3, that's the hydrogen that forms the bond with adenine. It's Watson-Crick pair. So in terms of chemical evolution, we don't want it to occur. And also, there's the hydrogen abstraction in nitrogen 1. That's also an important reaction site because that's where it is the bonds between the sugar, the ribones of the sugar, and the uracil to form the nucleoside. So these are the reactions between uracil and hydroxyl radical, and also we studied how does adenine affect hydrogen abstraction reactions and radical adduct formation reactions between hydroxyl radical and the uracil part of the Watson-Crick pair. These are the hydrogen abstraction reactions, and these are the radical adduct formation reactions. So how do we study the reactions? We studied step by step of each one of the reactions. We divided by reactants, reactant complex, transition state, product complex, and products. And we made some optimization plus frequency calculations to each one of these steps. And the computational details are that we use a cluster for high performance computing using Gaussian 9 on Linux. And as a graphical interface, we used GaussView 6. There's where we perform of the optimization and frequency calculations for like the reactants, reactant complex, transition state, product complex, and products. So additional calculations to study also in liquid state. In liquid state reactions, we made some solvent model density calculations, and to connect products, reactants, transition state, and products, we made some intrinsic reaction coordinate calculations. So what do we find out? If you can look, there are a lot of energies, but if you can look to the green energies first, we can see that the hydrogen abstraction in nitrogen 3 in a more diffuse system in gas phase has the highest transition state energy and also has the highest product energy. Then the transition state energy means highest transition state energy means that is the less probable to occur. And the highest product energy means that is the most unstable product from all the products. And this is why it's important to look at the nitrogen 3 reaction again is because that's the hydrogen that bonds with adenine. And with you can see the purple energies, you can see that the transition state from hydrogen abstraction in nitrogen 1 has the lowest transition state energy. That means that is the most probable reaction to occur. And also you can see in the product energy that has the lowest product energy. That means the most stable energy of the products. So in a reaction between hydroxyl radical and uracil in a diffuse system, the reaction that will happen the most or will have the highest yield will be the hydrogen abstraction in nitrogen 1. And how does adenine affect to this kind of reactions? It increases a little bit transition state energy, but also it lowers a lot the product energy. And a quick overview of what I just said. Here, nitrogen 3 has the highest product and transition state energy. Nitrogen 1 has the lowest product and transition state energy. And in both reactions with adenine or without adenine, it has the lowest product energies. So now let's go with radical adduct formation in a more diffuse system. If you can see, the reactions have very low transition state energies and also very low product energies compared with the previous ones. It has lowest transition state energies. That means that radical adduct formation in competition with hydrogen abstraction will have higher probabilities to occur, and with the energies they are kind of similar. So in thermodynamic conditions, it will be in a very high competitive system. And the effect of adenine, we can see it here better. We can see that when the hydrogen abstraction, radical adduct formation reactions have lower energy and lower transition state energies and lower product energies. That means that adenine likes to be around uracil because it increases the reactivity of uracil. And a quick overview, we can compare radical output formation reactions and hydrogen abstraction reactions. We can see that radical output formation have lowest transition state energies and also have lower product energies. So is more favored to radical adduct formation reactions. And also adenine lowers transition state energy for radical adduct formation reactions and also lowers the product energies for radical adduct formation reactions. So how does being in liquid phase affects the reactions of hydrogen abstraction? We can see here if you look to the green energy in the product energy of nitrogen 3, the reaction that for chemical evolution purposes, we don't want it to occur. It has the lowest product energies and also also has lowest energy in nitrogen 1 hydrogen abstraction reaction. These two are like competed, winning nitrogen 3 reactions. Why these have the lowest energies is because they are amide radicals and they are very electrophilic, so they like to be around water. And with these energies, we could say that it doesn't favor chemical evolution because nitrogen 3 reaction will have always the highest yield, but where does it balances is in the transition state energy reactions. We can see that in the purple energy, you can see that nitrogen 1 transition state energy there's a 10 kilocalories per mole energy gap between nitrogen 1 hydrogen abstraction and nitrogen 3 hydrogen abstraction. So there is the chance of chemical evolution in this energy gap. And the effect of adenine, it highest the energy, but also you can see that again nitrogen 1 hydrogen abstraction has the highest yield when it is alone. A quick overview: nitrogen 3 has the lowest product energy, competed with nitrogen 1, and also they're both with adenine or without adenine, they have the lowest product energy reactions. So another way to study, another way that we are studying the stability of uracil is how does it interact with minerals. Now we are studying with kaolinite. This is the structure of kaolinite. It is a 1-1 cell that's formed with a tetrahedral sheet of silicates and also with an octahedral sheet of alumina. So what we are doing is with a cell of kaolinite, we are taking part of the cell and study its interaction with silicate surface of silicate surface and also the part of alumina surface. So we are studying its interactions to calculate the energy absorption. And the first results that we are getting are here that when uracil is absorbed in silicate, it lowers a lot the energy to make it more stable. And with alumina, we are still calculating. So in conclusion, we use the DFT calculations, density functional calculations of optimization and frequency to study step-by-step hydrogen abstraction and radical adduct formation between uracil and radical hydroxyl. Hydrogen abstraction in a more diffuse system favors the formation of an amidal radical in nitrogen 1, and also in the same system, it doesn't favor the formation of amidal radical in nitrogen 3 in a more diffuse system in gas phase. Adenine lowers the energy of the products of hydrogen abstraction and also decreases the energy of products and transition state of radical adduct formation. And DFT calculations of solvent model density allow us to approach to track reactions under the influences of solvatation through optimization and frequency analysis. In liquid phase, both nitrogen-1 and nitrogen-1 amyl radicals are the most stable products due to their electrophilic properties. Silicate sheets, the first conclusions from adsorption is that silicate sheet significantly lowers the energy of uracil upon adsorption. If you want to send me some questions or something else, here is my email. And before the questions, I want to acknowledge institutions that helped me to do this research. CONACYT, Nuclear Science Institute, and my master's coordination, and also NoRCEL for giving me the space to show my research. And to acknowledge the PAPRIT project and also the
LUCA is the
Computational
middle
Center of
point
Nuclear
and
Science.
the final step of this complex process of chemistry evolution that start with the formation of the first star, the formation of the Earth, the acquisition of a lot of molecules, and the acquisition of water for the formation of the hydrosphere that permits the chemistry reaction for the formation of the probiotic chemistry. And in this process, the formation, the nucleic acids, amino acids, and each other. And after that, finally, the origin of light that is some controversial and exists a lot of theories about how the light origins. And well, the final step is LUCA. LUCA, how looks LUCA? Well, this is an artistical representation made by David S. Goodsell that represent in this image that LUCA is in a process of cell division, and we can see a lot of proteins that is present in actual cells. And another cellular parts, we can see the cellular membrane, we can see ATPase, and we can see the ribosome in process of traduction, and we can see the DNA. And well, we can note a lot of things of how looks LUCA, thanks for studies of which protein and which metabolic pathways is present in LUCA. One of the most recent studies about this is a study of Modi in 2024 that reconstruct some proteins that is present in LUCA. He reconstruct that LUCA have a complete machinery for the traduction, have the ribosome, have the amino acid, tRNA, synthetase, can synthesize all these tRNAs. He reconstruct a lot of metabolic pathways that is necessary for the life, the metabolic pathways for the glycolysis, for the synthesis of the nucleotides or the amino acids, for the production of acetyl coenzyme A with the metabolic pathway of Wood-Lunhan, and have the ATPase synthase and inclusively reconstruct some enzymes necessary for the replication of the DNA polymerase, and the DNA ligase. And Modi reconstruct a very complex LUCA. And exist other studies that have the similar results, like a study of Rapito in 2022 that reconstruct in the same way some similar metabolic pathways, but incomplete, like the metabolism of amino acids, carbohydrates, lipids, and each others. And well, with the knowledge about which proteins and which metabolic pathways are present in LUCA, we can know some things about the environment of LUCA. And one of the most recent scenarios about how is the environment of LUCA is that LUCA maybe is a cetogens or a metagens, and for the production of his energy, use the carbon dioxide and hydrogen that The source is the serpentination process in hydrothermal vents, and these molecules travel to the atmospheres and reach to LUCA for the synthesis of the energy. Inclusively, exists a hypothesis that LUCA can be can make the production of the first organic carbon source for other microorganisms that live with him or after him, and for the establishment of a deep heterotrophic ecosystem. And well, in the other side exists another theory that the LUCA maybe is autotroph, more similar to extremophils that live in hydrothermal vents. For example, the hypothesis made by Weiss in 2016, he proposed that LUCA live in extreme environment and use the geochemistry of the hydrothermal vents for the production of his energy using hydrogen, carbon dioxide, and sulfuric acid. And in the similar way that Modi made his hypothesis, have the Wood-Lunhan pathway, but Weiss reconstruct that this route, this metabolic pathway is incomplete, have less proteins in comparative to the Modi that is most complete metabolic pathway. And well, Weiss say that LUCA life in acid media and have the ATP synthase and have some essential metabolic pathways that, like the glycolysis for the production of his energy. And well, we know a lot of things about the environment of LUCA, which proteins, which metabolic pathways are present. But one of a lot of remaining question is, was LUCA an oligotroph or a copiotroph organism? And well, first, I will talk about the difference about these growth strategies, that is the copiotrophy and the oligotrophy. And well, we can see this graph and that represents this concept. In the x-axis, we can see the nutrient concentration, and in the y-axis, we can see the specific growth rate. And well, in the red line, represent the typical oligotrophic growth in microbes, and with the black line, we can see the typical growth rate of the copiotrophic. And well, we can see that the oligotroph have a less growth rate in comparative with the copiotroph, and we can grow in low nutrient concentration. And in the other side, the copiotroph need more nutrient concentration for growth, and the copiotrophs can grow more in comparative to the oligotroph microbes. And well, exist some traits that can say to us if a microbe is more similar to a copiotroph or a oligotroph. Some traits is the carbon source necessary for the growth and the generational time, but exist some traits that can say if an organism is a copiotroph or oligotroph, that are imprints in the genome. These traits are the mean genome size, number of tRNA genes, and the number of rRNA operons, the codon uses bias, and the GC content. And well, this graph represents this is the imprints in the genome. And for example, the oligotroph tend to have a mean genome size more short in comparative to a copiotroph organisms, have less number of tRNA genes and less number of rRNA operons in comparative to a copiotroph organisms, and tend to have less codon uses bias and less GC content in comparative to a copiotroph microbe. And well, why is interesting to know if LUCA was a oligotroph or a copiotroph microorganisms? Well, in one side is the theory that the LUCA maybe can live in a complex ecosystem with other microorganisms, and no exists only one LUCA. Exists a complex ecosystem of a lot of LUCAs, and well, the oligotrophy and the copiotrophy strategies in much times describes the dynamics of the population of these microbes. And well, in the other side, that is interesting know if LUCA is a oligotroph or a copiotroph is the hypothesis that the LUCA may be live in a near of the hydrothermal vents and is an autotroph microbe. And well, in the hydrothermal vents, the one of a lot of characteristic is that the environment is poor in nutrients and a lot of organic carbon source. And well, if we reconstruct that LUCA is a oligotroph microbe, can be help to explain or know something more about if LUCA maybe is a autotroph microbe. And well, the objective of this work is reconstruct the imprints in the genome, like the genome size, coding density, and GC content, and generational time associated with the growth strategy to estimate whether LUCA was more like a copiotroph or oligotroph. And well, the method is we use the Vieira Silva 2010 that is R213 species, and we used this data of this author because she made a recapitulation of minimum generational time of these 213 species in base of experimental data. And well, we download the data associated to these 213 species, and using the GTDB database, we recompile the GC content, the mean genome size, and the coding density. And well, we extract the 16S RNA sequence from the annotated genomes from the MCBIs. Using the tool SINUHE, we do the alignment, and after that, the selection of evolutionary model and the inference of the phylogeny was made using IQ3. And well, after that, using Bayesian methods, using the tool Bayes-Trace, we made the ancestral reconstruction characteristic of the coding density, genome size, the GC content, and the minimal generational time that is present in LUCA. And well, this is the results. The field trait reconstructed is the minimum generational time. And well, we obtained that the mean of this minimal generational time reconstructed is around of the 39 hours. And well, some microorganisms that tend to have this generational time tend to be oligotroph. And some examples of these microbes is Wigglesworthia glossindea, Treponema pallidum, and Buchnera aphidicola. And well, in base of this trait reconstructed, we can say that LUCA maybe is a oligotroph microbe. And the another trait reconstructed is the genome size. We obtained that the mean genome size of the reconstruction is of 2.5 megabase. And well, in previous studies, reconstruct the mean genome size of LUCA. And well, for example, of this is the study of Vaiduri in 2021 that reconstruct that the genome size in megabase of LUCA is around of 2.4 megabase. And in the more recent studies, like the MODI 2024, reconstruct in the same way that the genome size is around of 2 megabase. And well, example of these organisms that have this genome size exists Archaeoglobus fulgidus, Bifidobacterium longum, and Caldivirgia machilensis. And well, this mean the value in the genome size typically correspond to a oligotroph microbes. The other trait is the GC content. We obtained that the mean percent of the GC content it's around of the 30%. And well, never before had been reconstruct this trait in LUCA in the same way that the minimum generational time. But exists some hypotheses about the GC content of the LUCA, but no is so conclusive and specific about LUCA. For example, the Gospodinov in 2020 proposed that during the establishment of the codon user appeared codon less rich GC content, and in the moment of the LUCA appear, maybe have the less GC content in comparative in comparative to the origin of life. And in the other side, the most near approximation about the GC content in specific in only one gene in LUCA is the study of Basal in the 2008 that proposed that the rRNA had a GC content at around of 78%. And well, don't exist a consensus of global GC content present in LUCA or in his genes, but exists some information about this trait. And well, the GC content reconstructed correspond to a oligotroph microbes. And for example, of this is Mycoplasma genitalium, that is a oligotroph microbe. And in the other side, exists some copytroph organisms that have similar GC content, like Bartonella quintana. Well, the last trait reconstructed is the coding density. The coding density is around of 83%. And where exists some evidence that the copytroph have less coding density in compared to the oligotroph microbes. One example of the study of this trait is made by Chirac in 2023, and he do a metagenomic analysis in freshwater and marine pelagian environment and freshwater from the sediments. And he do a measure about the coding density present in these microbes. And well, the freshwater and the marine pelagian is an environment that the characteristic is that is poor in nutrients in comparative to the sediments. And well, we can see in green and blue that corresponds that the microbe of this environment, that they have a more coding density in comparative to the microbes present in the sediments that have a less coding density. And well, in this case, for this trait reconstruct, correspond more similar to a copytroph microbe. And for example, of these organisms with this coding density exists Buchnera afidicola, that is a oligotroph, and one copytroph that have this coding density is Bacillus thuringiensis. And well, finally, the conclusions and the perspective of this study is that we need to examine the additional genotypic features associated to oligotrophy and copiotrophy, such as the codon bias and the number of tRNA genes and rRNA genes. And our findings suggest that LUCA possesses a greater number of oligotrophic traits than a copiotrophic traits, Nevertheless, additional research is required to refine our understanding of Lucas growth characteristic. And in form of summary, a table about these reconstructed traits. And we can see that the minimal generational time, genome size, and GC content is more similar to oligotroph microbes, and the coding density is similar to a copytroph microbe. And well, acknowledgements to NoRCEL for this space, for this opportunity to talk about my work and the work of the Dr. Luis Jose de Lalle. Thanks to the SIMPE staff and the LikeBio for all the help, and thanks to my partners of the laboratory, and thanks to CONACYT for give me the money for the study of the master degree. And thanks for your attention. Here is my email for any question that you have.
Daniel HELMAN. I'm presenting a project called Ice on Early Earth as a source of energy for redox reactions. We'll go over ice strain, electricity, and protometabolism. This work was done while I was a visiting assistant professor at Wofford College in Spartanburg, South Carolina. First, let me thank my research assistants, Truman Sabank from Governor's School of Math and Science in South Carolina, and Ellis Parsons and Graham Seegers, both from Wofford College. They were absolutely wonderful, really great to work with. And let me thank also Wofford College for their grant that allowed me to do this, allowed us to do this summer work. Okay, so let's start with some background. I want to recall to mind the 1871 quote from Charles Darwin, talking about a warm little pond. That life may have started in a warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present. And that calls the question, why don't we see new life forming now in warm little ponds? And there's some answers for that, but right now I'm just going over background. 1979, we have the deep sea Alvin exploration of the Galapagos Rift, and see some deep ocean microbial communities. And this brought some interest to these settings as a potential place for abiogenesis. There are great chemical gradients, temperature gradients. They're rich in different chemical species. But it also calls the question, for example, Trixler's work, if RNA is too dilute, how will it autocatalyze if it's just there in seawater, or even in the rock? And let's go ahead a little bit to 2020 with Damer and Deemer, their work in geothermal fields. Rather than being at mid-ocean ridges, we have a setting in a volcanic regime with aliphatic compounds coming perhaps from meteoric showers, serving as membranes. But again, it calls the question, RNA, if it's going to autocatalyze, it needs to be very concentrated. Okay, so that's some background. Let's jump back a little bit and look at these missions to the moons of Jupiter and Saturn. And the 1995 to 2003 mission to Jupiter's moon, Europa, as well as the 2004-2017 mission, NASA mission to Saturn's moon, Enceladus, brought some really great evidence that these icy moons were underlain by oceans. You have some views of cryovolcanoes with geysers of water coming up. And so here's really when the idea of a snowball, like a icy worlds hypothesis, starts to take off. People see these icy moons and they say, oh, perhaps this is the locus of abiogenesis. And so this is one of the key components of my talk today, is looking at an icy worlds hypothesis and seeing what kind of mechanism might have been involved in abiogenesis. Okay, so let's jump back a little bit again to work from the 60s showing that there was low latitude glaciation, meaning glaciation on Earth in the tropics. And these are two periods, both Precambrian. One is in the Neoproterozoic, about 0.7 billion years ago, and one's in the Paleoproterozoic, about 2.5 billion years ago. And these aren't evidence from high latitude. They're evidence from low latitude, and so the inference is that the entire globe was covered with ice. And let's see where this is today. There's a challenge here in that microfossils are older. You find Archean microfossils from about 3.5 billion years ago, and then isotopic enrichment, suggesting early life, all the way back to 4.1 billion years ago. So this is the state of the science right now. Perhaps early life came from another place, panspermia, or perhaps it developed on Earth through abiogenesis right here. Okay, so at about 4.1 billion years ago, the average distance of the moon to the Earth was about 1.7 times closer, which meant that the force of gravity would have been about 3 times as strong, 1.7 squared, and tidal acceleration would have been about 5 times as strong. Earth rotation rate, you have, let's see, Earth rotation about 10 hours, 10-hour days at that time. So you would have had a tidal effect every 5 hours or so. Lunar inclination was a little higher, perhaps around 15 degrees rather than 5 degrees from the ecliptic. And what this means is if the large glaciers overlying some region were subject to tidal forces, perhaps some energy from that strain, that stress, might have contributed to some early chemical processes. And if we want to be careful in looking at what chemistry might have been there, we can look at living organisms and see that there's an abundance of phosphate involved, both in phospholipid bilayer for cell membrane and also in metabolism. And you have carbon and nitrogen species, perhaps from a carbonatite type volcanic setting, apatite phosphate dike. Overlying glacier may have ground down the rock, and the RNA, if there was a nucleobase or nucleic acids, may have been concentrated in the ice. So if the glacier is subject to tidal forces, It's also subject to strain heating, which is basically just the product of the stress and strain. We also have, let's see, potential for redox reactions. You have chemical gradients, thermodynamic conditions, and with a voltage, we'll get free electrons that can move from oxidizing to reducing agents. And that's in fact what we see. Kulesa et al., 2003, did some wonderful work at a glacier in Switzerland. They put some sensors down a borehole to the base of a glacier, and this top graph you see, it's mostly diurnal, daily self-potential of about 300 millivolts. And if you do a Fourier transform, now the top is days 25 to 40 in 1997. The bottom is showing Fourier analysis. The K1 is at 1 day, S2 is at 2 days, the middle tick is day and a half. We see a strong lunisolar daily signal, and also a strong solar diurnal, so twice day signal. We don't see M2, we don't see a diurnal signal correlating with the moon. What that suggests is that rather than gravitational, this self-potential is from most likely atmospheric effects. The twice per day is probably from maybe the electrical effect of solar influx into the ionosphere and atmosphere, maybe producing some induction. So let's look a little bit towards ice physics. The most mobile charge carrier in ice is not the electron, it's the proton. What that suggests is if you're going to do something electrically with ice, that changes of pH are the pathway to look for. In organisms, we see proton pumps, so this is not so unknown in living organism. And we have, in fact, I've done some work with a colleague, Matt Ritalik, and we made batteries out of ice by putting some acid in between two different layers and had some dopants. And we got about 0.1 milliwatts for several days. So it's definitely electricity that we just generated. Ice also makes a very good dielectric material, and it's paraelectric. Let's talk a little bit about what that means, paraelectric. Here's an idealized picture of an ice crystal. You can see it's hexagonal. The black is meant to represent oxygen, the white hydrogen. So you can see this is an H2O molecule. The white here is coming out of the screen. And this one, you could see the white is going into the screen, or you imagine. So you have this alternation. You have the 3 hydrogen coming out and 3 hydrogen going in. Now, for a ferroelectric material, that is a material that has electrical poles that you could switch in the same way that you can switch the magnetism of a ferromagnet. You'll have 2 competing lattice unit cells, 2 different ways to think about the organization of the material, and an applied electric field will change the charge, the electric charge that's expressed. Ice is not ferroelectric, but it is paraelectric. This is related to the lattice cells. A high pressure form of ice is in fact ferroelectric, but as it as the form is a little different, what paraelectric means is that the material responds in a nonlinear fashion to an electric field. So this makes for great capacitance, wonderful capacitor material, and has some other electrical properties too. You get some expression of electricity as you change temperature. You also get electricity from strain. So you get about 2 × 10⁻12 coulombs per Newton. That's about typical of most piezoelectric materials. Okay, let's see. Here's a video of some of the experiments we did. Hopefully this will play.
Well, they're not as good as like legging drops.
So you can see the trace over here. So that video showed, here, let's see. You could see the trace of the electricity that we were getting. We had a block of ice, about 2.5 kilos, with additives, typically about 5% by weight, combination of chloride salts, sulfate salts, carbonate salts, phosphate salts. One time we added a citric acid to a mix, just to see. And we were looking to see how much electricity we'd get. The top results, we got about 0.1 volt for sulfates. The very top results were from phosphates, about 0.2 volts. And the rise was about 0.2 volts per second. So that's about the amount of electricity we're looking at. Sometimes when the block is melting, we get about 0.1 volts just from the melt, but typically chloride and carbonate inhibit the voltage, whereas sulfate and phosphate were pretty good about producing it. Let's see how much time we have. Okay, so we did some energy calculations by attaching some resistors of known resistance in parallel, and we got about 10⁻5 watts from the water with phosphate, and the energy of about 10⁻8 watt hours. But the water itself, if you don't dope it with anything, you'll get about 10⁻3 watts and 10⁻6 watt hours. So that's the scale of electricity we're talking about. Let's look at some traces first. Here's where the rotation starts. Here's where ice flies off. This is a trial we ran with magnesium sulfate. And you can see as it flies off and slides along the floor, it's discharging just like a capacitor would. That's the same kind of discharge trace that you'd see in a capacitor. This kind of sigmoid here is where the test leads were removed. So that's an artifact of the measuring device. It's not the ice. But you see here a nice discharge like a capacitor. Here again, you see, here's the rotation start, and then here it flies off, and you see nice voltage. This is potassium aluminum sulfate. And the voltage correlates definitely with the acceleration. Sometimes acceleration has very little effect, like in this block, which was a mix of sulfates and carbonate and chloride. You see there's almost no effect, and the scale here is in say a few millivolts. Sometimes as the block is thawing and there's more liquid, you'll get a larger effect. We'll get some induction as well. This little blip here is from adjusting the block of ice by hand, sometimes just putting hand on the base below the ice. We'd see some kind of induction effect. So definitely paraelectric effect is active, at least from what we saw. Okay, so again, this kind of sigmoid signal is from attaching the test leads, and again, we'll see it here from them being detached. Here, the block flew off and then fractured. So part of this signal is from flight, and then part is from the test equipment after the test leads get detached. One can imagine like fracture of ice in situ, creating some kind of charge discharge at some point as well. Some of the readings we got were very noisy. This one was connected with a 1 kilo ohm resistor. See how noisy this trace is. This is calcium phosphate. This really didn't happen like this unless there was a resistor connected, and unless it was the phosphate. Phosphate seems to be, phosphate in ice seems to be pretty reactive here. Okay, let's see. Our results suggest that ice may have played a role in protometabolism. And there may have been local regions of periodic chemical activity. We have a draft manuscript in preparation called Protometabolism on Icy Worlds, Preliminary Experimental Ice Strain Energy Data and Their Implications. And for future work, we're looking to do a Miller-Urey type experiment to see possible chemical evolution in this kind of icy worlds place.
Thank
Thank
you
you
all very
so much
much
for your
for
attention.
attending this event. I would like to thank NoRCEL's for giving us the opportunity to showcase the advances of our research. My name is SINUHE, and I will be speaking to you about my topic, Vaalbara's legacy in the ancient earth, geographical continental evolution, and the probiotic chemistry. This is a subject we are developing at the Institute of Nuclear Science at UNAM in the Laboratory of Chemical Evolution as part of my degree work with the support of my professor Alejandro Heredia. Let me introduce you to the topic. First, a general overview of Vaalbara and its importance in the early Earth. Vaalbara is one of the Earth's oldest supercontinents, formed between 3.8 and 2.8 billion years ago in the Archean Eon. It was composed of cratons like the Catball in South Africa and Pilbara in Western Australia. Geological and paleomagnetic evidence suggests that these cratons were united before separating due to plate tectonics. This continental evolution, starting with Vaalbara, not only shaped the Earth's geography, but also influenced a fundamental process of the probiotic chemistry. This process of continental evolution, which began with Vaalbara, would not only shape their geography. The tectonic impact on probiotic synthesis is this occurred particularly in fault and breed environments where rock fractures and friction generated free radicals and hydrogen peroxide, as described in some studies of triboluminescence and mechanical free radical formation. The igneous and metamorphic rocks contain molecular level defects, particularly oxygen in a valence state of -1, which are usually dormant, wherein the rock's crystalline structure is differs as activity when the rock undergoes mechanical stress, such as in tectonically areas of during fracturing. Studies have shown that stressed rocks behave like a battery, generating a potential difference between the stressed and unstressed parts of the rock. When mechanical stress is applied, bonds between the affected oxygen atoms break, releasing positive hosts or chain carriers, which are essential missing electrons in the crystalline structure. These positive hosts can travel through the rock, acting as an electric charge flow. Positive hosts reaching the rock-water interface become highly reactive oxygen radicals. These radicals interact with water molecules at the interface, extracting hydrogen atoms from the water and forming hydroxyl radicals. Two hydroxyl radicals can combine to form hydrogen peroxide. Uh, this reactive oxygen species may have played a crucial role in the evolution of early anaerobic metabolic processes, creating microenvironments conducive to the prebiotic synthesis of essential biomolecules such as proteins and nucleic acids. The prebiotic chemistry and the mineral absorption. The absorption and polymerization of amino acids on mineral surface, especially on minerals like feldspar, graphite, and olivine, were a favorite process in the early air conditions, which chose uh, these minerals for laboratory work because Vaalbara's stratigraphy consists almost entirely of mafic to ultramafic volcanic rock and some felsic volcanic rock. Olivine is a common mineral in mafic and ultramafic rock, being rich in magnesium and iron, while plagioclase feldspar is abundant in igneous rock, especially volcanic rock of intermediate felsic composition, and also in some mafic rock. Graphite is rare in volcanic rock, as it usually forms under metamorphic conditions rather than direct igneous processes. For example, some rocks can contain olivine, for example, basalt, comatite, peridotite, and some rocks can contain feldspar plagioclase, rhyolite, basalt, dacite, tonalite, carbondiorite. And in the case of metamorphic rocks, can form due to change in the earth crust. Metaformic metamorphism occurs where existing rock, igneous, sedimentary, or even other metamorphic rock experiences high conditions of pressure, temperature, or the influence of chemical fluids, leading to change in their mineralogical and chemical structure without melting completely. These changes typically happen in dim crustal regions, but can also occur near the surface in areas with intense tectonic activity. According to computational and experimental studies, minerals catalyzed biopolymer formation by reducing the energy barrier of condensation reactions. The absorption of amino acids on siliceous minerals and feldspar facilitated the formation of peptide bonds, providing a chemical pathway for primitive protein formation. This process is part of the rock polymerization theory, where minerals not only absorb biomolecules, but also catalyze their polymerization in aqueous and possibly gaseous environments. The interrelation between the tectonic evolution and the probiotic chemistry. Uh, the Vaalbara's legacy involved not only the creation of primordial terrestrial geography, but also has implications for molecular evolution. Regions where tectonic plates interact actively provided a favorable environment for the production of these reactive species, which are case agents in prebiotic chemistry. Since the deep oceans of the Hadean were under high hydrostatic pressure, they offered a more established environment protected for harmful radiation than the Earth's surface, which has highly unstable due to meteor bombardment and other factors. The relative geological stability provided, uh, for example, less, less exposure to catastrophic impacts, protection from extreme water events, more moderate tectonic updates, stable conditions over geological eons, and a stable geothermal process. Uh, Additionally, oxygen species generated by rock fracturing may have influenced chemical processes that led the probiotic molecule synthesis. This suggests a coevolution between tectonic process and prebiotic chemistry, where tectonics not only shaped Earth physically, but also provided the chemical catalyst necessary for life. Um, as As part of the research we aim to achieve, we are conducting computational simulations where are assembled a pair of amino acids, L-alanine, B-alanine, and lysine, on the crystalline surface of three specific minerals, olivine, feldspar, and graphite. The goal is to understand molecular interactions to model how amino acids interact with mineral surface under a specific conditions, pH, temperature, pressure, that might have existed on early Earth. This helps to understand how minerals may catalyze peptide bond formation and facilitated the polymerization of amino acids to form primitive proteins. Identifying mineral catalysis with computational models can predict which minerals we more efficient in absorbing amino acids catalyzing biomolecular formation. This is essential for identifying what type of mineral surface favorite the synthesis of prevalent prebiotic compounds. Uh, Integration of another uh, result is the integration of the tectonic with the tectonic theory with the probiotic chemistry. In this context of Vaalbara research, the simulations to demonstrate how mechanical stress in tectonically uh, acid environments favorite the absorption of amino acids and the formation of reactive species of oxygen by modeling conditions in rock fracture zones or water-rock interface. We can explore how this geological environment influences the chemical evolution of biomolecules. Additionally, we aim to validate the hypothesis of co-evolution between tectonic and chemical processes, observing how amino acids assembled on minerals may have been modified with the active tectonics and this reactive oxygen species generation in the environments like reef conduction area. And thank you all very much for your valuable attention. I will gladly to share more results from the article we aim to publish as soon as they are due available.
And I would like to thank NoRCEL for providing this opportunity. I believe this is a very good opportunity to hear from others and hear various theories and experiments. I really enjoyed this. And I'm going to be talking on the possible roles of ionizing radiation forming the ROS environment of early life. I'm also involved with the International Big History Association, and I work at Argonne National Laboratory. And my email is there and will be at the end also. And so what I'd like to do is just a little bit of an outline of what I'd like to discuss. The first is radiation and life. Usually is what concerns me in my work. I usually work with nuclear technology and then also the impact on the environment. And so I've seen a lot of things, and I really wanted to explore this particular area. And so after radiation and life, we go to reactive oxygen species and radiation. So some of the reasons for considering them and some of the possible contributions of them. And so the first possible contributions is the direct generation of the reactive oxygen species. But also there are other things, for example, generation of electrical chemical potential. And for example, there are alpha voltaic batteries or cells that can kind of generate the potential. Obviously, when there's radiation, there's charge separation. And there's a lot of electrochemical potentials out there. There's also the development of mechanisms for repair. Other people are looking into that. And then the generation of energy in deep microbial communities. So we'll talk about that in just a little bit. And then I'd like to take a look at two specific kind of models for where life might have originated and see how it fits in there, if it's needed, and what people have concerned themselves with this topic. And one is a nuclear geyser model, kind of a more recent one, but one that more explicitly looked at radiation and generation of oxygen species and chemicals. And then the alkaline hydrothermal vent, mostly considering Nick Lane's metabolism first research. So let's go ahead and look at radiation and life. So living organisms have quite a capability to repair background damage of radiation. And this is applicable to my work all the time because we work with nuclear cleanup. And we really don't know exactly the effect of very low dose radiation because, radiation has been a part of life for a very long time. And so there are various repair mechanisms. And the thing is that there's still radiation all around us, and it's mostly natural, from radon in the air to external radiation from the ground to cosmic rays, hitting us from space. And even though we have radiation around us right now, it's actually quite a bit higher early part. Can you hear okay? Can everyone hear? Okay. So we have radiation provides high energy ionizing radiation that can promote energetic reactions. And so not only the ROS, but also energy things like hydrogen. It also provides charge separation for where you need a potential. And we've seen other talks that looked into, the various ways that potential can be generated. It also occurs in volcanic magma rocks, and then later seen as the rocks kind of age in things like granite that are used for building. And we definitely see high radiation levels kind of coming from buildings built with granite-like materials. And a recent experiment which looked at what happens if you remove that background radiation from biological organisms. And so it's very tough to find conditions like that. You have to remove the cosmic rays, the ground, the radon. And so they found, a deep mine and looked at it for two different types of bacterial growth. And when you remove that background radiation, you try to reduce it as much as possible, it does affect or degrade the bacterial growth. Does not stop the growth, but it degrades it to some extent. So it's a very fascinating topic. A lot of people are looking into it. There's obviously a lot of money being spent on cleanup, and we would like to know a little bit more about that. So a lot of very different topics there, but it's very difficult to study because you have to wait a long time and the results are statistical. But there's also a potential source of energy for subterranean life. There's 2006, there's a hypothesis of microbial community far below Canada and South Africa. And the belief there is that it was, radiation that can provide some of the chemistry and some of the energy of that microbial community. Just the last quote on there is that one can conclude that all sources of energy able to start formation of reactive species are potentially friendly to the origins of life, also possibly in other places of the universe. And I think that's what we've been seeing here. A lot of people have different ways of looking where the energy could come from. And so I just have some diagrams on the right showing the energy that fuels a deep biosphere. And so we know kind of things going on right now that radiation really, is believed to affect the chemistry and the energy sources of these microbial communities now. And so the question is, what kind of level was it, importance level was it early on? And the other one shows kind of the various contributions of radiation to us now. So we have the radiation in life. So now what about reactive oxygen species and radiation? And so there's two kind of common technologies that are kind of important right now that really depends on this, and that is a lot for cancer treatment and radiation. And it's actually the radiation that causes these ROS kind of chemicals, causes oxidative stress, and that's how many of the cells kind of respond to that radiation due to the extra energy and the chemicals going on. And so, there is some physical things kind of going on, but I think other people have kind of talked about them in generating various species, and people can do models to calculate, over the different types of processes going on, physical to chemical, how the species kind of evolve. But this is also very important for nuclear reactor corrosion, and that is there's a lot of water around nuclear reactors for cooling and energy transfer. And there are obviously some ROS being generated in that water. And so people really have to make sure that that does not cause a whole lot of corrosion. So they're very careful about the type of materials that are used, and a lot of research kind of going on in that area. So this is just a intersection of what's kind of people have studied and possibly how we could look at this in terms of origin of life questions. So the next one is, what are the Earth's stellar radioisotopes sufficient? And so first of all, the question becomes, we talked, a lot of other talks here have looked at other sources of high energy. For example, ultraviolet light, lightning, Miller-Urey experiments, cosmic rays available. All those are kind of available at the surface. But what about subsurface environments? For example, hydrothermal vents, which is deep ocean. You're not going to get a whole lot of UV, lightning, or cosmic rays down at that level. So that's kind of the environment that I was kind of thinking about. And so there are potassium-40 and uranium series in the Earth, kind of from the stellar explosion that helped create the solar system that's still in the Earth. And the question is there's still enough radiation there? Well, if you think about it, it's still heating the interior. About half of the Earth's heat flow is from the radioisotope decay. The other half is from the original gravitational collapse. And that drives plate tectonics. And so it's moving all of us, not too quickly, but it's still there moving us. Still very important for geological processes. There's also was much higher radiation levels earlier, and including the uranium was more enriched in uranium-235, which is the fissile isotope used in reactors and also in weapons. And so this becomes important when we look at the nuclear geyser kind of hypothesis. So the question becomes, if we do need high energy input from radiation, is it enough just to have the natural magma, or is a natural nuclear reactor as proposed in the nuclear geyser hypothesis? And so is it, or is a high energy source even needed? Are there other ways of generating enough energy to overcome some of the barriers and activation energies for biological materials? So I show the two potential sources of radioactivity. One is kind of in the Earth, based on the last solar kind of explosion, or last few solar explosions that led to formation of the solar system. And then the nuclear geyser kind of hypothesis. And in fact, there has been documented that nuclear reactions can occur naturally. One occurred a couple billion years ago in what is now Africa. And there was a certain situation that occurred where the reaction took place. It occurred for about, I believe, 500,000 years. Obviously not explosive, but enough to generate heat. So first of all, the nuclear geyser hypothesis, this is kind of more recent. You can see down below that in a book, Conflicting Models for the Origin of Life, that was published last year. They had one chapter in it, and it's by a group from Tokyo Institute of Technology, Moriyama and Ibiyasuzaki, and they also had earlier papers on this, and they listed 9 requirements for origin on Earth life, and they looked at a potential from the warm little pond of Darwin to panspermia, transport from Mars, hydrothermal vent, hot spring, and geyser system, and with their 9 requirements, they ruled out everything except the geyser system. Well, I think there would be a lot of discussion about that, but I have not seen too much so far on that. But I believe some is being discussed, and I'd like to look into that a little bit further, especially what's being said about the hydrothermal vent that looked at for Nick Lane. And so what they have is the nuclear geyser type of model. And so the next page kind of continues with what they looked at. And what interested me is that they were really highly focused on the energetics of the chemistry, saying that, look, if you have nitrogen, carbon dioxide, and water, you can see the energy levels of it. And it's very tough to get to intermediate products to actually generate some of the organic type of materials needed for early life. But if there is some other energy source that keeps it kind of out of equilibrium with ROS type of molecules, then things kind of flow a little bit more, and it's a little bit easier to generate some of those products. And one of the main reasons that they had was to free up the nitrogens, triple bond in there, not necessarily the oxygen, which we would look at from the radiolysis of water. And so, as I mentioned before, there was a natural nuclear reactor that had occurred, and that is because there was more enriched than uranium-235. But it still required some special circumstances and possibly life to facilitate. in terms of the precipitation of the uranium and the concentration of the uranium ore in a certain place. But they do kind of put this model together of the nuclear geyser system, and the nuclear geyser is just one part of it, with the uranium ore kind of heating up kind of the reservoir of the water that leads to the geyser. They have the cyclic system, so it's kind of a drying and wet kind of condition that leads to polymerization. They also have some things very similar to hydrothermal vents, where they have various forms of iron in there. For the hydrothermal vents, it's mostly iron sulfide that's in there. But they have obviously a little bit more complicated system than the hydrothermal vents. And so they have this actively going on, and they believe that this satisfies the 9 criteria that they have. And so the question becomes, exactly how much radiation is going from this uranium ore into this reservoir and creating these particular conditions. So that's one thing that I need to take a little bit more into. So I'd like to compare this with kind of another hypothesis that's been going on with hydrothermal vents. And Nick Lane obviously has been looking at a lot of these questions. I'm most familiar with the vital question, why is life the way it is, where he goes over a lot of his recent research, but he also has books specifically on oxygen, which I think would be kind of important for this topic that we're talking about. Also, the deep chemistry of life and death, and very early one on the origins of life. But for that particular hypothesis, it is kind of in the deep ocean. And it's not, he focused not on the black smokers, which are very thermally hot, but more on the white smokers, which would be off to the side. And so these would be alkaline hydrothermal vents, very reducing kind of conditions, so not much in oxygen there. And so with sulfates and carbonates kind of coming out in there. So different chemistry from the black smokers, which are the hotter ones and the ones which people focused on quite a bit earlier. And so he has, focused on these and just a little bit more on that. And so Lost City was one of the white smokers found in December 2000 with Alvin submersible. That produces hydrogen and methane, along with some more complex bioprecursors, and also provides some ammonia, which has the nitrogen, although not exactly clear where and how that is being produced. But they have come a long way at simulating some of this chemistry in the lab. So I have one article there, but it's pretty much, as mentioned kind of before, serpentization of rocks, kind of like olivine, in that type of environment where there are thermal and chemical gradients, along with some energy involved. And so the question becomes, is that enough energy to actually generate all these various species, or is there still some barrier that had to be overcome? And so Nick Lane's approach has been metabolism first, and looking at separation by minerals, so iron sulfide minerals, and very thin layers kind of separating 2 flows of the water that generates a potential. And as many others have mentioned, that the potential is very important because it's a proton gradient that is seen in most of life. And so this generates a proton gradient in here. And so there is some energy kind of in there, along with some of the minerals, iron sulfide, which is kind of seen in some of the biochemical materials and catalysts also. And so both of these sides are reducing environments, one more reducing than the other. And so, again, oxygen not too prevalent in this particular case. So he goes and develops, various ways how that chemical environment might proceed, first being contained within the pores of the space, and then eventually some of the membranes being developed and being free to go out. And so it's a highly reducing environment. The nitrogen from ammonia in the vent. Others have argued that there's not enough energy for the reaction. And of course, Nick Lane and his group came back with an answer for that, or hypothesis for that. And for the oxidation type of chemical events, they consider catalysts and molybdenum as an oxidant. And so they have various papers out there and are continuing to work on this. And so what I'd like to just say is that just taking a look at these two kind of hypotheses and looking to see where this radiolysis from ionizing radiation might come in. And so I believe that I need to look further in that criteria that was brought up with the nuclear geyser model, and what they raised against the hydrothermal vent model, and other kind of criticisms or questions about these models. And then another question is whether there are alternative high energy reactions. For example, in Lane's model, which is very deep in the ocean, not near the typical sources of UV lightning or cosmic rays. And we also have to look at the calculation of uranium enrichment for planet formation, and others have done that. They started very early when they first, found out the formation of uranium, but been updating it with calculations, not only for the Earth, but for planet formation at various times over the course of history in the Milky Way. And the thing is that uranium-235 is definitely more enriched earlier. And so there's different levels of heating in planets as a function of time, and also of size. And then kind of the one that I'm more concerned about is the low dose radiation effects. Really what is going on there, and how do these biological systems kind of react to this? And the other question is, how long has that radiation repair mechanism been going on? And did it first form from radical oxygen species, reactive oxygen species, or was it from the radiation itself, or was it kind of from the oxygen species created by the radiation? So a lot of questions out there. I just wanted to say, thanks for listening to this. I hope people will email with questions if you don't get a chance to ask them here. Again, thanks to NoRCEL for this opportunity. International Big History Association. I'm currently the vice president of this, and I would invite others, if interested, to look into Big History Association, which looks into origin of life and obviously other parts of the history from the Big Bang to the present, and then also Argonne National Lab, where we're trying to figure out what actually happens for these low dose the effects of low dose radiation.
All right. A very good morning from the beautiful city of Iowa, United States. Myself, Krishnendu. I'm a postdoc fellow at the Carver College of Medicine, Department of Pathology. And I'm a cancer biologist by training and an astrobiologist by passion. So first of all, please accept my sincere thanks to all the panel members and especially the NoRCEL for allowing me to present my topic in this esteemed platform. The topic which I'm going to present today is purely my own brainchild. It's a kind of hypothesis that I have recently started to work upon. So although I know this is only a very, very primitive portion, very, very new start, and there are a lot of polishing needed to make this hypothesis really happening in the bench work and followed by a publication. So I think in terms of science, the most relinquishing question is what is life? And that can be answered in many ways, in terms of philosophy, in terms of science, in terms of chemistry, and whatnot. But apart from the minute difference in between the living and non-living, the same question arises, what does it mean if someone says that someone is living? To put forward in a different way, NASA has proposed a beautiful kind of definition of life, a self-sustaining biochemical entity that would abide by the Darwin mode of evolution. And not only that, something which can actually abiding by the central dogma of genetics, that means from DNA followed by the RNA in the means of transcription followed by the proteins, because we know the proteins are the building blocks of life and each and every kind of phenotype that we usually see. So taking notes from this thing, the first question is how the life originated. It should not be the earth itself, because we know there is a vast universe outside. So how life originated in earth and elsewhere in the cosmos? And at the same time, are we really alone in this universe? Well, this is the beginning of the fascinating field of astrobiology. So today I'm not talking about this portion. I'll be much more focused on this one. Whenever we come to justify the various theories and various kind of conceptualization to justify the creation of life and how life originated, there are number of hypotheses and theories, starting from the spatial creation to the spontaneous generation, panspermia, hydrothermal vent theory, and whatnot. But apart from these theories, there are some speculation. Yes, I have to say this because these are speculation. Why these are speculation? Because these are depending upon some kind of science that is merely speculative, because we haven't been able to discover or invent any kind of time machine still. So if we can in future, then we can see what actually happened billions of years ago. So there are articles that actually suggest that how life created and what were the ingredients for the creation of life. The basic building blocks of life, the pentose sugar, the amino acids. We are all aware about the famous Urey-Miller experiment and how the primitive life, primitive form of Earth gradually catered into the perpetuality of the life itself. Various kind of hypotheses are there. The RNA world hypothesis, the lipid world hypothesis, and more recently, the RNA peptide world hypothesis. There are a lot of them. At the same time, another fascinating history that actually, I think, catered the life system on Earth is this great oxidation event and the inception of the cyanobacteria. Because before that, the oxygen level was very low. With the inception of this cyanobacteria, the great oxidation event occurred, and that increased the amount of oxygen in the atmosphere. But that particular great oxidation event had another impact on certain microbial life, because before that, most of the microbial life, they were anaerobic in nature. So this great oxidation event had a deleterious effect on these microbes. So there was a complete annihilation of a big chunk of the microbe at that point of time. And later, those who survived, they modified, they adapted to some new kind of genetical architecture for their survival. At the same time, so what would I want to tell if I need to add two different aspects of the early Earth. First, the abiotic condition, like the volcanism, the UV bombardment, the mineral cycle, weathering, this kind of thing, along with the scenario just after the great oxidation, where the level of oxidation increased in the atmosphere. Together, there would be the possible increase of the reactive oxygen species. Now we all know this reactive oxygen species containing various kind of highly charged oxygen molecule, including the superoxide, peroxide, this kind of thing. But what if this reactive oxygen species is going to work on the living system, or to be very precise, on the living system that were present at that point of time during the evolutionary process? Current literature suggests that there are a number of deleterious effects of this reactive oxygen species in the cell, from the cell death to deregulated autophagy, lot of inflammation, the cellular aging, epigenetical alteration, everything. So my question is, how this high level of reactive oxygen species actually toll upon the life on that point of time when the life just started to build up, and at the same time, what were the needs to cope up this high level of oxygen species? The answer is very simple. What we actually need at current scenario, loads of antioxidants. These are the free radical or the reactive oxygen species scavenging molecule that will scavenge on the reactive oxygen species, the free radicals, and that will decrease the level of reactive oxygen species lower within our cell. Now, depending on this thing, my question is, if I want to model this whole scenario, that means how this elevated level of ROS actually shaped the survivability of ancient life form and how they adapted to this extreme ROS enriched environment, I thought we should be having kind of model system, because from a biological perspective, I can say whenever we are going to do some biological experiments, we should be needing some model. That may be murine, that may be the reptile, that may be the amphibian model, that may be the ferret, that may be the rabbit, anything. But what kind of model should we be coming with to justify this particular hypothesis? So on one fine morning with a cup of coffee, I was just thinking upon this thing, and suddenly I thought, why not to go with the cancer cell? Now, we know that cancer cells are pretty omnious in nature. The cancer cells are having different kind of phenotypical and genotypical alterations, starting from the increased level of proliferation, high level of metastasis, immune destruction system and immune escaping system, epigenetical alteration, everything. But even after that, I want to focus my attention to use this cancer cell as the model system. There are plenty of literature that suggests that there is a very elevated amount of ROS in the cancer. The cancer cell is a kind of ingenious in nature that would actually use this ROS system for the next level of survival. Yet, high level of ROS is definitely going to kill the cancer. So from this diagram, we can see that our optimum level of ROS, which is definitely higher than what we usually find in the normal cell, is pretty much tumorogenic in nature. That means it can contribute to the formation of the tumor. But if the ROS level is elevated at a greater extent, that would actually be cytotoxic, and it can even kill the cancer cell. So what the cancer cells are coming up to cope up this increased level of ROS? The answer is again the ROS scavenger. There are a number of molecules that are present within the tumor stroma. Obviously, they are present within our cells too, but in case of cancer cells, they are much more elevated. Why? Because cancer cells are much more evolved in nature, and for the target to make itself survive in this huge area of high ROS level, cancer cells usually have a higher expression of this ROS scavenging molecule. Among these particular ROS scavenger, I'm pretty much interested in a particular system. The system is known as the thyroidoxin system. Thyroidoxin is a kind of component and belongs to a kind of scavenger, the ROS scavenging molecule system that includes thyroidoxin itself, thyroidoxin reductase, which is a kind of Selener enzyme, and with the help of NADPH, thyroidoxin plays crucial role in lowering the ROS level. What it does, usually, say it's a protein, and this protein is somehow gets highly oxidized. So the thyroidoxin portion, the thyroidoxin with the help of thyroidoxin reductase and with the help of the NADPH, it actually converts the oxidized protein into the reduced one. And now the protein is quite capable to show all its biological function, unlike the oxidized portion. So this is the way by which the oxidized protein, especially in the cancer cell, get escaped and later converted into the reduced protein, which is essential for carrying out number of functions. This is also happening in the normal cell, but at an elevated level in the cancer cell. And that actually caught my attention. Why not use this particular system as the model to justify how ROS shaped the evolution of the life in early Earth and how LUCA evolved to later? And one more interesting thing that I would like to introduce here, that this thyroidoxin system is highly conserved. That means the genetical architecture is pretty much conserved from the mesokarya to eukarya. That means if we can actually study this particular system in terms of their genetical architecture, we can be having a kind of model. Obviously, there are some literature that suggests if we knock down this particular thyroidoxin system in cancer, that will increase the death of the cancer, that will increase the radiosensitization of the cancer. So, in therapeutical approach or in anti-neoplastic condition, this is a well-established thing that this thyroidoxin is pretty much needed for the cancer cell survival. And I'm here to use this particular approach in a different direction. So one might ask this question, okay, why not consider any other normal cell or any other non-neoplastic cell? Because cancer cells are having certain kind of mutation, they're kind of different from the regular cells. So why I'm much more focused on the cancer cell? Because I can definitely use some bacteria, but cancer cells, they are eukaryotic in nature. And to me, the cancer cell is the best example of showing the elevated level of Darwinian fitness. That means if I want to mimic an experimental outcome where I need to bombard a cell with a lot of ROS, if it is a normal cell, it will eventually die. And I'll be unable to see that what kind of genetical changes are there to cope up the increased ROS condition because the cells are dying. But if it is cancer, it's going to play differentially. Certain genetical mechanism, like just I mentioned in my past slide, the thyroidoxin system, it is going to get elevated. Then what are the genetical system that will get elevated, that will get highly expressed to cope up this condition, will be an interesting thing to find. And at the same time, these cancer cells are having the long-term senescence. They don't get into the senescence condition pretty early. So we have a long term of time to see the various effect of ROS on the cancer. It's much more easier to culture the cancer cell. We can directly culture various kind of cancers in terms of cell line. At the same time, we can directly culture the cells from the patient sample to see what actually is happening in the tumor microstroma. Cancer cells, they're not actually foreign in our body. Unlike any other bacteria or virus, they are not foreign in nature. So it's the kind of rebellious cells that are present within our own system. So this kind of thing might be an essential tool to justify why I chose the cancer system. And not to mention that we can induce cancer in any other species by various kind of biological system like the xenograft, like the syngenic. We can simply induce some portion of the cell line into mouse or any other organism to induce their tumor, and then we can study how those tumor cells are behaving differently than that of the peripheral natural cell, especially in terms of high elevated ROS system. So these are the foundation of my initial hypothesis that I'm currently working upon. Obviously, it will be needing a lot of polishing, and I have started to talk with some of the eminent person in this field so that we can have a plan and we can go with this kind of thing. So currently, the last thing that I want to mention, that the cancer cells are now being used in the field of space medicine and astrobiology. Recently, the cancer cells are being incorporated in various kind of study in the International Space Station. At the same time, the cancer cells act differently in the microgravity condition. There are some ongoing research to see how the microgravity, the space radiation, they are working upon the cancer cells and how the cancer cells are behaving differentially. So there are certain approaches currently going on where the cancer cells are being used a kind of model. So this is the final frontier of my initial hypothesis, to use the cancer cell to check how the transformation from the last universal common ancestor to the last eukaryotic common ancestor happened. Usually we know that there are different kind of cellular compartment formation, various kind of de novo synthesis, various kind of other kind of microbial association, endosymbiotic theory. But in terms of ROS, in terms of survivability among high level of reactive oxygen species, how the life got its shape, we can do this thing, taking the cancer cell as a readily available, sustainable model to justify this. And with that, I would like to rest my talk here. Thank you again to NoRCEL, and thank you so much for this lovely audience, for your patience. And these are some of the glimpses of this wonderful Iowa city from where I belong. And it's the UNESCO World Heritage Site. So I'll be ready to take some of your questions.
Well, it's a pleasure to be here and attending this meeting, exciting meeting. And thank you, Johan and NoRCEL for the invitation. I think it's wonderful. So it gives me an opportunity to show a little bit of our research. So let me see if I can eliminate this. Yeah. So I'm going to, I will be, let me see, yes. Okay, for some reason it's going very slow, okay. So I'll be talking today about the birth of structural domains and intrinsic disorder in protein evolution. I start with this slide, which is it's an exposition in the Wellcome Library in London with this neon art. Kind of illuminates the challenges that we have with the structure of proteins. And for many years, I've been exploring in my laboratory the evolution of proteins. And what we do is basically a back-in-time exploration. We use an ideographic historical retrodictive framework. It's also known as a top-down approach instead of bottom-up approach to understanding origins of whatever. In this case, I'm interested in molecular biology, and my interest is to understand the complexity, the molecular complexity of life. And for that, we realized a long time ago that the three-dimensional structure of molecules provides a window into function and evolution that is very deep. And that is basically the approach. During this more than 20 years, we've been exploring the evolution of genomes at different levels. And we started, you know, in 2003, there were only 32, believe it or not, genomes that were sequenced from which we could extract the proteomes of them and study and provide a global view of the universe of protein architecture, as we call it at that time. And with time, we've been adding genomes to the analysis, and this is just a sample of some papers out of more than 100 that were published. At some point, we settled in about 8,000 for a simple reason, is that at the beginning, we had, our limitation was the sequencing effort, the genomic revolution. Now the limitation is phylogenetics and the ability to basically generate trees. So as soon as you are over 10,000 you know, leaves of a tree, then you have computational challenges ahead. So what are the questions that I'm going to address today? Well, first of all, I'm going to try to explore with you the birth of structural domains of proteins. These are particular units that define the fold of a protein molecule. And it turns out that we have thousands of these folds available to us. And the question is driven by the need to explain molecular vocabularies, because each of those structures has functions embedded in them. There are specific mappings of functions to those structures. For instance, the Tim beta alpha barrel fold that you see there has 34 SCOPs, superfamilies. SCOP is a database, a gold standard of classification of proteins. And it has 34 of those, and of course, multiplicities of variant domain families embedded in them. So now think that we need to understand that fold plus an additional 1,256 folds that have been, that we know exist, with their about 2,000 superfamilies associated with them. The other question that I have for you today is, what is the origin and evolution of intrinsic disorder? So when did the intrinsic disorder appear in evolution of peptides and proteins? And the question is driven by the need to understand why there is disorder embedded in the order of proteins. And to illustrate this, I'm showing this NMR solution structure of a non-structural protein from the SARS-CoV-2 coronaviruses. This is a crucial protein that interacts with NSP12, which is the RNA polymerase, and together with others form a replicase complex. Now, when you look at the solution structure, you have 20 structures aligned to each other. And you see that there's a region there which is completely disordered. You can see that there's not really good match at the alignment level, you know, in the structures. And then you have different helices forming a bundle that is very consistent in the analysis, showing that this is an ordered structure, three-dimensional structure with helical components with it. Now, one thing that you can do is to understand this molecule better. You can actually dissect that molecule into component parts. And these component parts are loops, and you can see different loops there. The red one is the one that holds the RNA binding property of this particular molecule. But look at the N-terminal helical element, which is highly disordered, and it has very atypical helices assigned to them. So the question is, why is it that we have that disordered region in the molecule? What is the role, how it has evolved in evolution, and so on? So those are the questions. Now let's start exploring how to analyze this. Phylogenomics involves finding elements to study, basically, in the structure of proteins, this structural phylogenomic approach. And usually for that, we need protein building blocks. And these protein building blocks are what we call prior molecular states. These are intermediate forms that mediate the growth of evolving macromolecules. They're sets of autonomous, integrated, coordinated, and stable interacted parts that we call structural state that needs to be individuated by dynamic behavior and cohesion. So this is a systems biology definition. But from an evolutionary point of view, whatever we study, these features need to be evolutionarily conserved. They need to be persistently dynamic so that they can justify their prior existence. And here you have different hierarchical levels of structural organization in proteins, from going from lower granularity, from higher granularity with amino acids, the peptide chimers, loops, and then reaching structural domains and above, basically, to form the highly complicated complexes that we have in a cell. And so we will focus on, in my talk, on loops and structural domains. But as you can see, there is an implicit timeline embedded in it, meaning that the time of origin of all these elements, you know, follows a somehow chronology, I would say, you know, and that kind of chronology is what we are basically after. Now, in our research, we've been focusing on how those elements, those prior molecular states or building blocks, combine to each other to form all that complexity that we need to study. And here, for instance, we studied how the individual domains of proteins combine with each other to form multi-domain proteins. And we've done this analysis at a network level, you know, understanding that complexity. We also were interested in molecular functions. And it turns out molecular functions interact with catalytic residues that are fundamental to interact with different elements. It could be other proteins, or it could be small molecules, to produce all the molecular functions of the cell. And we have dissected this too by studying these elemental functional loops that embed this. What I'm going to talk today is more about loop recruitment network, the idea that the loop structures somehow hold the elements of molecular function in a protein molecule, and these elements have to combine with each other in evolution to form that complexity that I showed in terms of structures. So how do we do this? We turned phylogenomics into a chronological process. In our Black in Time exploration, we are trying to unfold those building blocks, and for that we need a global algorithm to study this. And this algorithm works in the following way. We need to find evolutionary conserved features in genomes, our data, establish a ground plan and model of change, a model of evolution, track back in time, retrodict by optimizing data and evolutionary change onto possible ground plans, and then building series of time events describing the history of the biological system and subsystems, and then trace that history on the systems that we study, let's say a molecule or RNA or complex or whatever. So basically that's the sequence: structure, evolution, phylogeny, chronology, networks. That's kind of what the algorithm does. And here you have a workflow, a simplified workflow. So we start with a genomic census, we generate a phylogenetic data matrix, and that data matrix is used to build a tree of parts of whatever we study, and a tree of wholes of what we study. And then from the parts, we generate ages of those parts, the time of origin of the parts. And from those times of origins, we build chronologies and then trace those chronologies of parts in wholes. And these wholes can be proteomes, molecules, functions, parts can be domains, substructures, whatever, So this is a generic way to do this. And again, here you have, for instance, an example. The entities that we study are the protein domains in proteomes, and we can generate a tree of proteomes in a tree of domains. Here we have a tree of proteomes where we're studying wholes. And here the tree of proteomes includes viruses. It's a tree of life, ultimately. And as you can see, there are two elements here, two ancestors in the nodes or branches, internal branches of this tree. One is LUCA, the last universal common ancestor. The other one is the last universal cellular ancestor. We have basically two elements there to study that we can dissect. But we can also generate the tree of parts. And here we have a tree of Koth architectures. This is a high-level fold characterization from the Koth database of proteins. And you can see, an origin. Two elements here that are interesting I want to focus. One is at the base of this tree, which is rooted, you have sandwiches and bundles, like the one I showed you earlier. And the other thing that I want to show, midway in evolution of this tree, you have a particular Koth architecture that is related to proteins that are completely disordered. They're called intrinsically disordered proteins. And they come late in evolution. So intrinsic disorder through this tree appears to be a latecomer, something that appeared late in evolution. So let me just go through quickly how this retrodiction exercise with structural domains works. For instance, here we analyze 8,127 proteomes from archaea, bacteria, eukarya, and viruses. It turns out that those proteomes hold about 3,192 domain families, which you can visualize through that Venn diagram that you have there. Remarkably, in the Venn diagram in the center, you can see those 979 families that are universal, meaning that there is a trunk in whatever tree is embedded in this data. And of course, there you have an example of the domain with what we are studying. And we can construct a data matrix seen here as a heat diagram, where columns are structural domains and rows are proteomes. And what we do is basically, from this phylogenetic data matrix, we construct a tree of domains on the top. And this tree of domain has the property of organizing that data matrix in a particular way as a chronology, because the tree is unbalanced. It has that property, that very peculiar protein and powerful protein that allows us to build chronologies out of these phylogenetic reconstructions. And therefore, that matrix now is organized with a flow of time. Domains are ordered according to the time of origin, that is their emergence in evolution. Now, some time ago, we published a molecular clock of folds, the existence of a relationship between biomarkers related to geology and the actual structures. And it turns out that there is a very strong correlation between those two elements. So we can basically, thanks to that, a relative scale of time turn into a chronology based on on a time frame in billions of years. So given this, we were able to kind of trace one by one all the individual structures that we have as leaves on these trees. And it turns out that there is a gradual appearance of these Venn groups in time. So if you can see here, you have colored the tree with the branches of the tree with what we name as evolutionary phases, beginning with phase 0 and ending with phase 5. And it's a clear progression of those Venn groups of the Venn diagram. And this has been incredibly consistent with all the trees that we've reconstructed during the years. We have a phase 0, which is the rise of the last universal common ancestor, a phase 1, which is the rise of the last universal cellular ancestor, a phase 2 that defines the reductive evolution in archaea, a phase 3, which embeds the emergence of bacteria, a phase 4 with the rise of modern viruses and diversified superkingdoms, and finally a phase 5 with expansion of higher eukaryotes. And this has been consistent from a point of view of the time of origin of individual leaves that we study, but also in terms of the kinds of mappings that we've been doing on top of these chronologies. So the chronologies, that chronology that I showed is a chronology of domains. But we can build chronologies of protein loops too. So these protein loops, we consider them as being stepping stones to protein structure. So these are more granular elements or units of evolution that are below the level of the structural domain. And how do we define these loops? Well, there's a database, it's called ArchDB, that uses a number of features of a loop, the bracing structures of the loop. You have an helical element and a strand there in that loop in the left. And so that defines the type of loop that you have, and we have 10 of those. And then we can use a length of the individual structures, but also a number of angles that define the actual topology and geometry of the loop. And we can use this to classify all the loops that exist in proteins. And it turns out that, that we've defined 88,000 of these prototypes, which are groups of loops bound together by the same geometrical and topological properties. And those prototypes can be mapped into about 5,000 non-modular loops and about 2,000 modular loops, which we can map in the timelines and generate the networks that you have there. So we map loops to domains, and as you can see, the modular ones, a loop is present in many domains, so therefore you have a number of lines interacting between loops and domains as they unfold in time. And the non-modular ones appear only singly associated with individual domains in the timeline. So, so the wonderful thing about this is that then these evolving networks that we have mapped in evolution provides a description of the emergence of protein domains by the combination of loop structures. So we have, with these networks, a way to understand how is it that a particular fold appeared by combination of individual loop components. And it turns out that, we've seen in this network two big waves of innovation. One initiated by the P-loop structure, and the other one by the wing helix structure that embeds the wing helix domains. And they all appeared very early at the time of last universal common ancestor and Lucella. And most of the types appeared very early in the timeline. There you have the 10 different types that are possible of loops, and those are individual prototypes that I'm showing to you, which are the first ones that appeared in evolution. And they all appeared very early in that timeline that you have below, all in phases 0 and 1. Here you have the two prototypes that are the founders of these two waves, the P-loop wave and the electron-Alex wave. The P-loop wave embeds this particular prototype that I'm showing there, which is the oldest of all prototypes that we have in our phylogeny. And all this unfolded in 400 million years of evolution, beginning at 3.8 billion years ago. And those waves were fundamental because they are responsible basically for all initial metabolic reactions that appear in the protein world. Now, one of the wonderful things of having this kind of chronologies is that now we can, let's say, take one individual structure. This is the oldest domain that we've identified. It's called the ABC transporter ATPase domain-like structure family. They're classified with those concise classification strings that you have there, C37.112. It's just a classification from the database. It tells you that is that fold. And since we know the age of the individual loops, then we can trace the age of the loops onto the structure of any protein. And there you have that transporter with the individual components mapped with different colors. You can see the most red is the oldest, the P-loop in the center, followed by two additional domains that are kind of a mapping to different parts of the molecule. We can do the same thing with the first domain that initiates the wing helix wave. And again, here we have the nucleic acid binding electron-Alex domain. Remember, this wave is related to binding of nucleic acids to proteins. And note how this appears later in evolution. So an indication again that nucleic acid interacted with proteins later in evolution, once complex structure was already present on Earth. So the wonderful thing about this also is that we can now dissect this evolutionary patchwork of loops by using artificial intelligence. And now this fantastic deep mining tool that we have available to us that is called AlphaFold 2, which is a modeling system that, given a sequence, allows us to produce a structure with quality that is the prediction is as good as an experimental prediction. This is what AlphaFold 2 does for us in these days. This has occurred since 2000, so it's a very recent tool that is available to us. So we've been using that to basically use the sequences as they are unfolding in evolution and see how those sequences are going to be producing the actual structural core in the middle there, level of the fold. And you have the P-loop structure on the top and the helix wing domain on the bottom. And you see each step there that is occurring, every several millions of years adds an additional loop to the structure. This loop kind of grows in time. And look how at 3.2 billion years ago, we have now a primordium of a structure where you have a sheet element being built, packed against a helical element. And then you have the structural core, which is the typical or the design of this particular P-loop fold that we are studying here. And it appears basically at 3.1 billion years ago. So it took a lot of time for the small little loop structure accrete into this growing molecule to form the structural core at 3.1 billion years ago. So, so this evolutionary patchworks that I was showing to you before, because of this ability, turn into processes of fold emergence that we can visualize as these folds occur. So this structural core that I was telling you about is this alpha beta alpha sandwich structure typical of the binding cassette of the ABC transporter, which is the oldest fold that we've identified in numerous of phylogenies over time. And so what you can now do is take that fold and use a tool that's called DALI that explores the all possible neighborhoods of proteins that exist in the protein universe and request information about the structural core. Remember, this is an hypothetical entity. It doesn't exist in nature. And remarkably, when you do that kind of analysis and explore the neighborhood of the structural core, what you find is that the closest structural neighbors are actually the folds that we are studying. So and that fold is there, the C37-112. Those are the structural neighbor, but then you see there another fold related to that, and that fold is the AAA ATPase domain family. So this type of analysis allows us to kind of dissect pathways of innovation, of structural innovation. So that structural core is basically a founder, not only of the ABC transporter fold, but also the AAA ATPase fold, typical of that domain family. So this has a lot of potential, to explore the evolution of proteins. And of course, then what we can do is, well, let's measure how this, molecule unfolding in billions of years of evolution is changing particular properties of that individual fold. And of course, one of the initial properties was to study disorder. What is the level of disorder of the growing molecule, that is evolving? And remarkably, we see that the initial loop is highly disordered, is within the high disorder category that you see in the plot below. And then that level immediately decreases to moderate disorder as the protein grows, and it maintains moderate disorder until it forms a complete structure at the end, which is the present day structure. And so there is this is indication, and this trend we see with many of these types of analysis, and this is just one example, we see a trend of disorder decreasing in evolution that we need to explain. So molecular disorder and size appear to be inversely correlated. So, so then we jumped into an analysis, a global analysis of intrinsic disorder in the protein world. And so we had domains, we had loops, we had chronologies of them, so we can map intrinsic disorder unfolding in time. And what we see in this mapping that you see below there, each individual dot corresponds to a loop that is unique to cells or shared with viruses. And I'm not going to describe the results that come from that kind of analysis, which are very interesting per se. But I'm using that just to illustrate that the complexity of the data that we are mapping in that chronology. And one thing that we see is that mean disorder is relatively high. So the vast majority of prototypes, of loops.
Gustavo,
yes.
2 minutes, please. Yeah.
Okay. The vast majority are highly disordered. And this pans out throughout the timeline. The other thing that we see is that prototypes are enriched in helix and strand bracing structures at the beginning of the timeline. but then they're replaced by other bracing structures. And those prototypes, the helix and strands, are structures that exhibit the lowest mean disorder level. So we can explain why there is this tendency towards increasing in disorder with time. Of course, we can look at the ancestors, LUCA and Lucella, and calculate the disordered level there. And again, you see the vast majority are highly disordered loops. What we find then is, well, this is another way to see this. We see that most loops that evolved were disordered, and they increase, that disorder, that number in time, in the timeline. And the ordered loops appeared later in evolution. They are latecomers. We did basically the same thing, which I'm not showing, with domains, and this is summarized by these plots on the right. And the thing seems to be reversed. Most domains that evolved were ordered, and disordered domains appeared later in evolution. So we are seeing a dichotomy here. Disorder manifested differently at loop and domain levels, suggesting to us that there is percolation of evolutionary constraints across levels of organization occurring. And this for us is really amazing, because we see this pattern of order evolving as a benefit to loops that were acquired late in evolution, but at the same time we see disorder evolving as a benefit to domains that were acquired late in evolution. This is telling us that there is a loop-associated short and domain-associated long regions of intrinsic disorder unfolding in time, manifesting opposing evolutionary tendencies. And the prediction here is that there needs to be a link between disorder and molecular size. This is the prediction. And so if we go to the extant world, the world of today, remarkably, we see that prediction unfold. Here you have the loops and domains, and both of them have shown inverse correlations between disorder and polypeptide length in, from a global point of view. Now, let me just point out very quickly that there is a lot of biophysical evidence supporting a correlation between folding time of proteins and size of proteins. And disorder is related to the folding process. You need to understand that. And so this correlation that we are seeing here is somehow manifest the folding process of a protein. And that folding process is basically unfolding this order disorder, equilibrium that needs to be, occurred during the process of protein evolution. So I'm closing here with some take-home messages. The beauty of phylogenomic chronologies, to study evolution from a global perspective. The fact that we can use that information to dissect the origin of viruses and cellular superkingdoms, and understand when is it that all these elements appear in this timeline of billions of years. Now I'm showing you that we are able to trace the birth of domain structures and make models of how those domain structures were produced in evolution, and how the loop, it provides a wonderful element to study the geometrical elements, and the biophysical elements of proteins and how they contribute to the formation of protein structures. And this incredible equilibrium between order and disorder that is unfolding in time. And I just stop here, and I thank NoRCEL again for the invitation, and Blue Waters for providing us petascale computing levels to perform all this analysis, and all the people that contribute, especially Fisa Mughal, Faiz Aziz, and my daughter Kelsey, which have been exploring this birth of protein domains with the AlphaFold system. And with this, I'm going to stop the share, and thank you very much for your attention. I appreciate it.
Okay. See you now, the next slide.
Yeah, it's moving.
Okay,
Yeah,
it's good.
we can see the
So
next.
it's about using water to solve the prebiotic water paradox. That title sounds a bit paradox, but I love such things. Normally, to solve the water paradox in prebiotic chemistry is to avoid water, and my talk is about just to use water to solve that water paradox. So the outline of this talk is I would give a short introduction about what this water paradox in prebiotic chemistry is, and I will introduce an easy way of its solution. And I will show this by an abiotic synthesis of RNA in just water, and it produces long strands with sequence complexity of such RNA. And the key role to achieve that is to use nano-confined water in particle suspensions. Why is this important? I think overcoming the water paradox via exploiting such nanoscale effects on water is prebiotic very plausible. It's both in line with geological ubiquitous settings, and it's in line with the conservative nature of evolution. So, what is the water paradox? The water paradox says that on the one hand, the water is of course very essential to life as we know it, but also it causes very unfavorable reactions with key biomolecules. This means, for example, the synthesis of nucleic acids from its building blocks, such as nucleotides. For this, the phosphodiester bonds which have to form are only formed in a condensation dehydration reaction. This is a chemical reaction that releases water. But the thing is that if you run that reaction within water, you have a thermodynamic barrier in that. So if the reaction product is very difficult to form when it's at the same time the solvent. That means in prebiotic chemistry, a spontaneous formation of nucleic acids is prohibited by water. So you see here in this scheme, if you have two nucleotides and want to link them via a phosphodiester bond, you see these two OH which become then expelled. So it's a condensation reaction which releases water. And as I said, if this reaction would take place within water, it would not occur. And this is the paradox in prebiotic chemistry. If you think that it happens in water chemistry, you wouldn't achieve such key reactions. For example, this polymerization reactions to make RNAs or polypeptides. If you take a look at the thermodynamics, we see that the nucleotide polymerization releases water, which means that we have a huge thermodynamic barrier. We have an uphill reaction, which is so in this way very inefficient to occur spontaneously. But the thing is, if the water activity is very low, then you can push this reaction into an hexagonic state, and this you can reach by reducing the water activity. It's what water can do with its environment. So the thing is, in the normal approaches to circumvent this water paradox, to reduce the water activity. And some approaches to do this is, just using alternative solvents, not water, or by just get rid of the water via evaporation, or adding condensing agents, or just setting very high temperatures to overcome that barrier. Well, if you apply such things to a modern living cells, the cell would just die. So there's a thing, is this prebiotic plausible? if we take a look at this, we have to consider two aspects. The one is, well, life manages the water problem within a stable environment, just full of water and at physiological temperatures. And the other thing is that we know from biological evolution, and it's probably the same with chemical evolution, that evolution builds on existing pathways. It's the so-called conservative nature of evolution. So, coming to the first one, life manages the water problem. Living cells, when we look at living cells that contain an intercellular aqueous liquid, which is densely packed with large, complex biomolecules. So we have virtually no free water, but only or mostly this interstitial nanoconfined water within a cell. And all the enzymes we have within a cell have also, especially this polymerase enzyme, which is making RNA, DNAs, have water-filled nanochannels within their active center. That means water exists mostly as in the form of nanoconfined water within a living cell. So what is nanoconfined water? And what is it, why it is so interesting? Nanoconfined water has very different properties compared to bulk. We have a completely different hydrogen bonding network dynamics. You have different densities, different reactivities, dielectric constants. And especially the phase diagram of water is shifted to gas-like. So in some you have, and many other things, you have a very anomalous behavior of water when it becomes nanoconfined. That's where the fun begins. We can take a look on what depends this anomalous behavior of nanoconfined water. Well, it's the very complex interplay of various nanoscale phenomena which are related to things like shear forces, surface energy, and size of the confining boundaries, the molecular structure, electrical double layers around that particles, and some other things such as the thermal Casimir effect and so on. So it's a weird setting. And if we then come back to the thermodynamics, and we have learned that by reducing the water activity, the thermodynamic barrier can be circumvented or overcome. The thing becomes interesting if you spotlight on nanoconfined water, because nanoconfined water changes the vapor pressure and the hydrogen bond network dynamics of water. These both two parts are exactly what reduces the water activity, well, as a function of gap size and characteristics of these walls, which are confined to water. So, in sum, if we see living cells, these intercellular liquid densely packed with large biomolecules, and we look at a cell from what I have studied, material science, you see nothing else than an aqueous suspension of nanoparticles, which generates temporal nanoconfined water. And then when you look, where can we find such temporal nanoconfined water in nature? You have a huge variety of nanogeochemical environments where we have that situation. We have sediments with pore water. We have hydrothermal vents with precipitated particles coming out of them. We have particle aggregates and water-filled lithospheric cavities, and we have atmospheric aerosols with particle burden. All these settings, we can have these mixtures of particle and water, and can have these interstitial and nanoconfined water. So then the central point, can this link between intracellular environments and nanogeochemical environments be interpreted as an evolutionary conservatism? Well, the thing is, this can be tested by addressing the questions: do abiotic nanofluidic environments within aqueous suspensions may have the potential of inducing key biochemical reactions? And we tested this, and we took an example reaction. This was the polymerization of nucleotides into RNA. And we do this in particle suspensions as a mixture of polyaromatic heterocyclic particles and inorganic particles. And well, our building blocks of RNA were nucleotides. Here's an example: adenosine monophosphate, AMP. We started with AMP because AMP is very interesting. You have AMP, or this AMP motif, in very other key biomolecules for energy and all the other things. And so AMP must be very important at the very early stage of chemical evolution. So we start with AMP and see if we can polymerize AMP. And we have made these nanoconfinements within particles and have these stacks of AMP you can see here at the center, and make also some force field calculations by them. I wanted to show you a movie, but it's not working here on this other computer. But you can see here on this image, we tested to make a dynamic molecular modeling. And here you have these confining walls of different particles and filled with water, and then these stacks of AMP. And we are interested if these AMPs would be, this is the gap size here is about 5 nanometers. If this AMP stacks would be stable or not, and we see yes, they are very stable, so they have the possibility to make these condensation reactions if possible. Okay, what we detected then afterwards in our experiments and our samples are mixtures of water with particles, inorganic and organic particles, and this AMP. And if we only have particles but no building blocks of RNA, we see nothing. If we only have AMP but no particles, we see something, a very low amount of RNA, but it's just below just beyond the detection limit, and it's caused by a statistical effect. If you have these densely stacks of AMP, you get indeed some RNA formation at every certain time point, but it's not much. It's background noise. But as soon as we mix both the particles and the building blocks, we see a huge amount of RNA compared to this, and it depends on which particles we use. I will come back to that later. And the other building blocks, UMP, CMP, are not so working so fine because their stacking capability is not so good as AMP. But if we put an amino acid also inside our mixture, which has by itself a very high stacking capability, it pushes also, we tested this with UMP, and catalyzes by stacking also to a higher level of RNA composed by UMP. So in sum, we see there's a huge difference if we put a dense particle mixture, if you use a dense particle mixture or particle suspension, it's called, and run our experiment in a dense particle suspension. Okay, we have this thing. We have another test with PCR, and also saw that we have lots of RNA inside. Even if after 8 cycles, this signal begins to start. So we have a detectable amount. And melting curves here, I will make it short. This is the sample. We see we put inside a known RNA with 19 nucleotides in length, and we see in the melting curves that they are shifted to the right, so they should be longer than 19. So we were interested, okay, we have RNA, but how long is that? We assumed that we have some micro RNA made, but when we applied gel electrophoresis, we were very surprised. We have lots of RNA, yes, but it's very, very long, which it's 4,000 nucleotides and even more long. So this was a really surprise. For comparison, we have other samples where we don't have our nucleotides inside, so we see virtually nothing. So yes, we made obviously some very long RNAs of poly A RNA, so it's only composed by the building blocks of A and P. So the next step was, okay, if we put all the building blocks together and make our particle suspensions, would we also see some RNA? And the answer is yes. We tested this with amorphous silicon dioxide nanoparticles. And we have some background noise with this silicon dioxide, but we have significant amount larger than that in our NMPs. So, okay, we have long RNAs. We also have RNAs from a mixture. So the next thing would be, do we have some kind of sequence complexity if they build sequences? Or do we have only AAA and UUU and CCC, or do we have a mixture of complexity? So we made a next generation nanopore sequencing. This was also a GUSTAVO, and see, yes, indeed, we have a very complex row of our AT and AC and things like that. So we have this doesn't mean that we have a meaningful sequence, but at least to get a meaningful sequence, you need a complexity in a sequence, and that we achieved. We can produce RNA with a kind of sequence complexity. So yeah, that's the thing. I will skip this, I think, a bit. We also tested if these nanoconfinement effects would be important for us from another point of view, from material science. We make scanning tunneling microscopy on molecular and atomic resolution level. And yeah, we'll skip this a bit. We see coverage of organic semiconductors, and we see a more coverage of organic semiconductors monolayer on particle surfaces when we add AMP, for example. AMP is doing something with our semiconductor deposition method, and we know that this semiconductor deposition method relies on properties of nanoconfined water. And when we remember that these other NMPs and CMPs don't increase the coverage, only AMP here in this experiments, and we also saw that in our RNA experiments, AMP produces lots of RNA. We can say that AMP do something with nanoconfined water, and in the other direction, nanoconfined water do something with AMP. So we infer that this differences in the extent of nanoconfinement phenomena between different samples correlates with the relative difference in amount of RNA. This indicates a synergy between nanoconfinement effects and nucleotide polymerization. Okay, so the first findings we can summarize. is that abiotic temporal nanofluidic confinements allow prebiotic RNA synthesis within water. Although this would be thermodynamically not possible, but if you mix this with this dense particle suspension, you get nanoconfined water, and it's possible. And it's possible under stable, just moderate conditions, the same conditions as within a living cell. So they emerge in aqueous particle suspensions, and such particle suspensions are geologically ubiquitous and thus prebiotic plausible settings. And the thing is that such settings are consistent with evolutionary conservatism, as living cells also work with temporal nanoconfined water. So we published that. You can, if you are interested, can download that paper. It's open access. And I add something to this talk today because the spotlight here was set on reactive oxygen species. So when Sohan introduced that, I became very interested in reactive oxygen species. I was not before. It was not on my radar before. And I saw that aqueous suspensions of micro and nanoparticles used for our RNA synthesis can produce substantial amount of reactive oxygen species, according to the literature. So, and according to the literature, I learned that nanoparticles are a major source of reactive oxygen species. So, nanoparticles are very important for reactive oxygen species. So how to make reactive oxygen species? Well, just add nanoparticles and make something harmless to something very different, reactive oxygen species, you can imagine. And what you have as a nanoparticle then is, we learned that substantial ROS forms in aqueous mixtures of mineral particles and ultrafine organic aerosols, according to the literature. And reactive oxygen species formed by decomposition of organic hydroperoxides. This is a very huge fraction of organic aerosols, by the interactions with water and reaction with metal ions. And what makes me interested is especially kinones in organic aerosols are redox active and are reported to induce reactive oxygen formations. And what we used before was anthraquinone, as you see here. In this slide here, this anthraquinone produces lots of RNA compared to others. And so we have a high yield in RNA. So that's where things become interesting. Originally, I chose anthraquinones because it's a prominent compound in carbonaceous meteorites. But now, when Sohan put the spotlight on reactive oxygen species, also from this point of view, it becomes interesting to use that. So, yeah, if you're interested, I can mail you also these papers then. And you see here in this slide that such nanoparticles, metal nanoparticles especially, can form via different ways reactive oxygen species, and also these polycyclic aromatic hydrocarbons, or these kinones, adsorbed onto these particles can then stimulate the synthesis of reactive oxygen species. So, and what I have learned from literature is that amorphous particles have a substantially more yield in ROS compared to other particles. And that also is interesting for me, because here in this slide, you see that we use amorphous silicon dioxide, and this was also a nice thing to use for RNA output. So I think I really like reactive oxygen species, and I will probably put it on my radar in the next experiments. Okay, so we come to the end. Once again, our abiotic confinements and low prebiotic RNA synthesis within aqueous particle suspensions, and they are geological, ubiquitous, and consistent with evolutionary conservatism. And the main message, the take-home message would be, don't be afraid of confined water in aiming to solve the water paradox. If you confine water, it will come out and make you surprises. And just dive to the bottom of the nanoscopic water world and discover fantastic things. So I thank my fantastic team of very different people who look from different perspectives onto these and make fantastic experiments. And I thank NoRCEL, which is a fantastic network. And gives me also
on behalf of my co-investigators, Eva Vergara and Jorge Valdez, I'd like to thank Sohan and others, or members of the organizing committee, to give us this opportunity to talk about something we're really interested in. Okay, so the focus of the talk, I want to pose a problem for you. The reactive oxygen species that we're going to be talking about require oxygen and they require energy to form. And yet, ROS appeared to have developed well in advance of the great oxidation event, and even before the so-called whiffs of oxygen. So how did this happen? That's the problem I'd like to pose, and I would like to discuss with you today. And then if ROS did occur, were there mechanisms and enzymes for mitigating the ROS activity? Did they also evolve very early, or did they come much later? And why is this relevant to astrobiology? So those are the issues I'd like to discuss with you. And just to put this into context, here's a background, which I think you've seen quite a few times already. And the question is, LUCA is back here around 4 to 3.5 billion years ago, and GO is around 2.3 to 2 billion years ago. So they're separated by at least 1 billion years. And yes, there were whiffs of oxygen, certainly before GO, probably by oxygenic photosynthesis. And yet, ROS are predicted to occur much earlier in the evolution of the planet, back way before LUCA, and antioxidants are predicted in LUCA. So is this a conundrum, a conflict? That's the question I want you to ask yourselves. So before we get into the details of this, I just want to add a few additional layers of complexity, because I will be coming back to these issues. First of all, there was a moon forming event right after the Earth formation, and the magma boiled, and so any life that would have risen very early was probably eliminated. And then the Earth began to separate by partitioning the lighter elements which floated towards the surface, and the heavier elements then which begin to form the core of the planet. So initially, there were cratons. These are small, primitive continental formations. And then they joined together and emerged as continents. But initially, there were shallow seas and probably not very much land. That's the first, I'm going to return to these points. And of course, initially, there was extensive volcanic and hydrothermal activity, which of course continues until this day. And yet at the very beginning, there were really violent volcanic eruptions, and probably the Earth was covered in hydrothermal vents. Thirdly, there were periodic meteorite bombardments, and I've indicated just a few of them here with the red arrows. And of course, they continued well into the Go and even closer today. And the position of the arrows I just put randomly to indicate that there were extensive, very large meteorite bombardments, especially before and just after Lucca. So I'm going to come back to those points. Okay, so there are 4 possible solutions to the problem. The problem, just to remind you, is why did mechanisms and enzymes, antioxidants, for mitigating ROS develop in advance of the great oxidation event? Well, 1, perhaps the dating of Go and Lucca is incorrect and they overlap. I'll address that very quickly. Then there's the evidence, perhaps, that ROS occurred in the oxygen oases. So it was not a question of atmospheric oxygen. It was oxygen that occurred in just very local places, way, way before Lucca. And then perhaps the prediction of anti-ROS genes and mechanisms in Lucca is incorrect. So I'll just take a brief look at that too. And then the 4th possibility is intriguing, that these antioxidant mechanisms arose to deal with other stresses, not oxygen, but other stresses, and then were repurposed or exapted to deal with oxygen later. So I'm going to mention all these 4 points. Okay, so first, is the timing of Lucca and Go correct? I'm not going to go into details here. The Go is very extensively accepted by the scientific community, and there's a huge, weighty body of evidence that the Go was between 2 and 2.3 billion years ago. I'm not going to discuss that. Lucca, there is evidence for Lucca. There's isotopic evidence. There's the isotopes of sulfur and iron and so on. There are microfossils at 3.5 billion years, and there are stromolites at 3.5 billion years. So life was definitely occurring at 3.5 to 3.8 billion years ago, right at the end of where I put the arrow for Lucca. There is also some very controversial evidence from zircon crystals from Australia that there was life at 4.1 billion years ago, and that's where I put in the beginning of Lucca. So Lucca is in that position. So although the timing of Lucca is highly debated, it can slide back and forth, there is a consensus that it occurred at least billion years before Go. So Go is fixed and Lucca is more or less fixed. So bottom line, that's solved. 2, were there ways to generate ROS and oxygen pre-Go? No, I'm not talking about the whiffs of oxygen that occurred, predicted whiffs of oxygen that occurred just before Go. I'm talking about deep generation of oxygen. And there are various ways that it can be done, and I've listed them here. And number 1 is the redox active minerals that we've been talking about, which occur in hydrothermal vents, and they are exuded by volcanic gases. And if you recall on the slide I have just shown you, there were extensive volcanism and extensive hydrothermal vents just before Lucca. Number 2, there's photochemical and ion radiation reactions. We heard about ion radiation reactions from David LePOIRE earlier today. And remember that I indicated there were shallow seas before the continents amalgamated and rose deeply above the sea. So those are both consistent locations for generating ROS and pre-Go oxygen. Number 3, meteorite impacts. Now, this is a new concept which we are developing. So I'm going to put it onto a separate slide, and that occurs now. Meteorite impacts, especially at the very beginning, were probably intense. So intense that the pressure and heat could alter the rocks, melt the rocks essentially, creating also hydrothermal activity, which could have then extended for thousands or millions of years. And they are known to release nutrients, for example, phosphate, into the oceans, and therefore perhaps feed nutrients to burgeoning life. And I've just given you a couple of examples. There are many, many more. Hematite, which is an iron-containing oxide and very common in the Earth's crust, when it's subjected to high pressure and heat, it will release oxygen. So imagine a meteoritic bombardment comes, releases oxygen, but only locally. And that oxygen is going to get used up, soaked up by other rocks and so on that need to be oxidized. But momentarily, there is perhaps free oxygen. And the decomposition of perchlorates. Perchlorates have been found on Mars, and when they are heated, they release oxygen. So I'll return to the Mars question later on. Okay, so the bottom line, there is no direct evidence for ROS on early Earth, but there is a substantial body of indirect biogeochemical evidence that indicate the presence of oases of oxygen in local areas and restricted in time. So it's not atmospheric oxygen, it's local oxygen. So number 3, the question is, were the enzymes and mechanisms to mitigate to ROS really located in LUCA? And that's a tough question because we don't have access to LUCA. So what people have done is a phylogenetic reconstruction, or many different ways of doing that, and using molecular clock techniques. There is strong evidence that LUCA contained, for example, superoxide dismutase. It contained catalases, perioxidins, and thioredoxins, and glutathione. These are all very well established ROS. There is some issue with this interpretation because, for example, if there is extensive horizontal gene transfer, HGT, a gene that occurs in a bacteria gets passed to an archaea, or archaea gets passed to eukaryote, or whatever, then it would look like the origin where they join together the root of life. It looks as though they both developed that gene independently, and they didn't. So that is a point of contention. So although there is good evidence that LUCA had the enzymes to mitigate ROS, and also the immediate descendants, so that was the methanogens and so on that emerged after LUCA, they also had enzymes and mechanisms to mitigate ROS. It's still a little bit contentious. Okay, what about the issue that I raised last? Number 4. Were antioxidants initially evolved to mitigate effects of other so-called antioxidants? And there are 2 very well-known ones. One is the reactive sulfur species, the RSS, and there are a couple of examples that I've given. And the other one is the reactive nitrogen species, RNS, nitrogen oxide and nitrogen dioxide. Now, notice that both of them also contain oxygen, but that's not free oxygen. So that's the difference between oxygen and free oxygen. There were possible prebiotic sources of RSS and RNS, and they're very similar to the prebiotic sources that have been postulated for reactive oxygen species, the volcanic emissions and lightning-induced processes, the geochemical and hydrothermal sources, the extraterrestrial sources, meteorites, for example, and interplanetary dust falling on the Earth all the time, and the UV and photochemical reactions in shallow surface waters, more or less very similar to ROS. So there is good indirect evidence that RSS and RNS occurred before or at the same time as the ROS were developed. So there is the possibility that genes and mechanisms arose to counteract RSS and RNS, and then was subsequently borrowed and developed, evolved, accepted to deal with ROS when the level of oxygen rose. So that is an excellent hypothesis, and it's an alternative hypothesis to ROS occurring at the origin of, before the origin of life. Okay, so what's the astrobiological significance? Now, there's a lot of significance, and I don't have time to detail all the sorts of things. I've just picked two to give you a flavor of how I think this is astrobiologically significant. Obviously, it's astrobiologically significant because it deals with the origin and development of life on our planet. However, what about other planets? So if you think about it, half the lifetime of Earth, it was anoxic, without atmospheric oxygen, although, as I mentioned, there is very good indirect evidence that there were oases of oxygen long before go. So this has an implication for the use of oxygen as a biosignature gas in the search for extraterrestrial life. We're going to, if other planets develop the way Earth developed, they're probably not going to have atmospheric oxygen, or if they do, only later in their life. So do we have the tools, the telescopic tools, to detect oases of oxygen? And the answer is probably not at this moment. Maybe something will occur later on. So we're going to miss a lot of potential habitable planets searching just for oxygen as a biosignature gas. And as you know, there are plenty of other potential signatures of occupation, for example, methane. So if you look at planets and moons where ROS are likely to form, and we know where they're likely to form: shallow water, hydrothermal activity, volcanoes, meteorite strikes, and thin atmospheres. The thin atmospheres would mean that solar and radiation could penetrate, and that could create ROS. Now, Mars is a perfect example. We know that Mars had small oceans at the beginning, and yet then it went dry. So during the latter part, it had very shallow water, perfect for ROS activity. And when that water dried up, there was also an immense amount of radiation that could have produced ROS and oxygen, and perhaps could continue to do that. As I pointed out earlier, perchlorate is quite common on the surface of Mars, and if heated, perhaps by meteorite bombardments, it could release a small amount and local oxygens. What about ice surfaces on Enceladus and Europa? Well, they are heavily bombarded by solar radiation on the surface, so they could form ROS there. And in the deep oceans, there's good evidence that there could be hydrothermal vents. So in this instance, what we're looking at on Earth does have implications for the search for life on at least other planets and moons within our solar system. Okay, let me just summarize the key points. I think there is strong geochemical evidence to support the ideas of early whiffs of oxygen long before go, It was likely localized, and molecular oxygen was produced in these hydrothermal events, shallow waters, volcanic eruptions. Likely that there were also early RSS and RNS and mechanisms and enzymes to mitigate their effects, and these were later exapted, used to fight ROS as the oxygen levels rose on Earth. Interestingly, these microenvironments, either ROS, RSS, or RNS, would have created selective pressures that led eventually to the evolution of oxygen handling mechanisms, setting the stage for GO and the evolution of aerobic life. And finally, I pointed out just a couple of examples of how this information assists interesting astrobiological lines of inquiry. Okay, what about the future? And again, I just picked a couple of juicy examples. Obviously, we need to further investigate the ancient rock record to identify additional evidence for pre-GO oxygen. We need to explore the molecular evolution of ROS detoxification in ancient microbial lineages, including the predicted LUCA. These are major projects and major open questions that require deeper analysis. Okay, so thank you very much. And now for the last slide, while I answer questions, if there are, call for papers is called Bridging Disciplines, Femophiles and Their Implication for Astrobiology. And I happen to be the special issue editor for this, and it's going to be published in a journal called Microorganisms, which has a reasonable impact factor, 4.1, not great, but not bad. And there's additional information if you want to submit a paper there, and the papers will undergo double-blind reviews as usual. And there is a managing editor, a managing person at the microorganism deal with it. So I won't be the reviewer. However, of course, it's always nice to have a friend.
Many models of prebiotic microsystems have been proposed that are considered to be precursors of the first living cells. For example, this is a model suggested by some Japanese scientists, giant lipid vesicle with incorporated DNA and some other molecular structures. The next step of its evolution consists in amplification of DNA because of oscillation of temperature. And the last stage of evolution is division of this giant vesicle along with amplified DNA. And then its further evolution stops. The same process is shown in pictures after 4 minutes, 5 minutes, and 10 minutes after experiment starts. And then further evolution of this model is over. Chemical composition of such model is similar to chemical composition of living cells. However, there exists a strong difference between prebiotic microsystems and living cells. First, all types of prebiotic microsystems do not possess the following key properties that demonstrate activity of living systems in the environment. First, the ability to boundless expansion, and second, purposeful behavior. The same Japanese scientists who suggested this model understand this principal difference, and they illustrated it in this picture On the left, main types of non-living prebiotic models, RNA, lipid, and protein, shortly non-life. And on the right, living cells, life, and between them, mysterious evolution jump or the missing link. Let's consider this border between non-life and life from chemical and thermodynamic points of view. This is the border. Above it, the root of the phylogenetic tree, eukarya, prokaryotic bacteria and archaea, extincted life forms, and below the barrier, main types of prebiotic microsystems, RNA-walled molecules, proteinoid microspheres, and lipid vesicle. From the chemical point of view, the both chemical evolution and biological evolution proceed in similar direction with complexification of organic molecules and structures. In this way, biological evolution looks like a natural continuation of chemical evolution. However, from the point of view of thermodynamics, the situation is different. All types of prebiotic, non-living prebiotic microsystems exist or evolve with a natural tendency to increase in entropy that corresponds with the second law of thermodynamics. But paradoxically, biological evolution proceeds in the opposite direction, with concentrating free energy as well as information. So, between life and non-life, there exists thermodynamic or entropic barrier. That is, its essence just consists in the inevitable tendency of non-living systems to entropy rise. So, it follows that any kind of prebiotic microsystem cannot be transformed into initial forms of life only due to chemical complication of matter. It is necessary to jump over the thermodynamic barrier to get living state. It is possible in the only case if a prebiotic microsystem obtains so huge impulse of free energy that compensate the tendency to entropy increase with plenty for some period of time. In this case, it is located above the thermodynamic barrier. Then, when the energy impulse is over, the system returns to the initial prebiotic state. And such impulse, the microsystem can obtain only under changes in the environment. That means under far from equilibrium conditions, accompanied with multi-level oscillations of physicochemical parameters. This state can be considered as intermediate state between life and non-life, and it is maintained in oscillating mode. Oscillations around the thermodynamic barrier. Otherwise, in other case, without such thermodynamic jump, transition to life is impossible because it means a violation of the second law of thermodynamics. The entropic barrier manifests itself in the existence of natural tendency for non-living systems to increase entropy. And the transition requires thermodynamic jump or inversion, entering a huge impulse of free energy into a network of prebiotic chemical reaction. This is that I told earlier. How can we indicate the thermodynamic difference between living systems where free energy prevalent over entropy and non-living systems where, on the contrary, entropy is prevalent over free energy? The basic formula to calculate free energy in physical processes is the following. It is relevant for physical processes and physical systems. However, when we try to compare such different kinds of systems like physical and biological, this formula is not relevant due to two reasons. First, we cannot mathematically strictly correlate entropy and free energy based on this formula, because free energy and entropy measures in different units. Free energy in joules, while entropy in joules to a degree. And the second difference and some more difference I will mention later. So, because of impossibility to quantitatively establish the border, thermodynamic border between life and non-life, I use more general terms: contribution of entropy instead of entropy, and contribution of free energy instead of free energy proper. In this way, the physical sense of free energy and entropy not changes. Free energy is a measure of high energy value, and it is a part of energy that can be converted into any kind of work. And entropy is the measure of low energy value and measure of disorganization. So the border, thermodynamic border between non-life and life, is defined through inequality, total contribution of entropy to total contribution of free energy in a system. And we can indicate three possible ratios. First, in non-living systems, this ratio or balance is positive. That is, contribution of entropy is prevalent. For living biological systems, on the contrary, this balance is negative, and contribution of free energy is prevalent. And intermediate state between life if contribution of free energy and entropy are similar to each other. Coming back to this picture, we can see that intermediate state between life and non-life is maintained in oscillating regime. Oscillations around the thermodynamic or entropic barrier. In the next slide, it is shown the border between life and non-life that is maintained is oscillating regime. In this system, free energy generates inside this system, and entropy also rises due to second law of thermodynamics. And in case free energy is prevalent, the system develops to life, to living cells in the future. And in case entropy is prevalent, the system returns to the initial prebiotic state. So the same is shown in this slide. Intermediate state, this is a border between life, non-life, or entropic barrier. This is oscillations around the entropic barrier, and two possible trends. In case the system is able to generate more and more free energy, it develops in the way related with biological evolution, or it can, in case this generating of free energy is insufficient to cover entropy, the system degrade and eliminates with the initial process of biological evolution. It follows of this that multi-level oscillation of physicochemical parameters are necessary for the origin of life, like three accepted conditions: aqueous medium, availability of organics, and energy source. And such oscillations in highly non-equilibrium medium is necessary to generate a huge impulse of free energy that allows to prevail over the entropic barrier. And the only medium that embrace all, that is characterized all four the required conditions for the origin of life, is hydrothermal systems. For instance, this is a list of organic compounds detected in hydrothermal systems in Kamchatka Peninsula, more than 200 organic compounds, including lipid precursors and nitrogen-containing organic compounds, which are biologically important. The second required conditions is multi-level oscillation of pressure and temperature in hydrothermal systems in Kamchatka. And this is irregular macro fluctuations of pressure accompanied with micro oscillation, more or less regular, in pressure. And even such regular oscillations in different fragments are very different, can be very different. For instance, this is fragments of about 4 hours, and we can see plain trend and more or less identical values of amplitude. This trend is plain, but very different amplitudes, and in this case changes the both amplitudes and some other parameters. This is correlation between temperature, concentration of temperature, and some other chemical compounds in hydrosomal systems in Slovenia, about 70 minutes cycle, and we can see that the peak of temperature more or less corresponds with the values, maximums of many other chemical parameters. So, in hydrosomal system near the surface, we can see prebiotic or organic microsystems, which are in very changeable medium with irregular fluctuations of temperature, pressure, and chemical parameters. These multi-level changes and fluctuations can stimulate interaction between these microsystems and with future, under favorable conditions, integration of it into the initial proto-community of living systems. Coming back to this initial slide, it follows that start of life and the transition to life of prebiotic microsystem to life occurred in oscillating regime, in oscillating mode. And this is initial starting point that should be exist also during the entire evolution of the biosphere. And actually, we can see in all present microbial population, we can see the periodic transition from active existence to anabiotic state. And this is circulation. First, when the microbial population is under unfavorable conditions, external conditions, begins to transit into anabiotic state. And first of all, there occur decrease in functional activity of the most complex structures like nucleic acids and enzymatic proteins. Then show down the movement of lipids, degradation of cells, and up to glassy state. This is why bacteria transforms into a resting cell. This is anabiotic state. And when the conditions changes to favorable, begins restoration of resting cell in the reverse direction from the most primitive, the most simple changes to more and more complex, and at the end stage arising a growth cell cycle and the formation of genetic apparatus. I would like to add that with small changes, this succession, this cycle is universal for all bacterial populations. And second, it is very well confirmed by plenty of experiments. And the hypothesis is that I propose is that first, this cycle of oscillations between active state and anabiotic state, it oscillates and it repeats starting from the initial life cells during the origin of first life cells. And in this case, we can use this succession of exit prebiotic systems from anabiotic state to the active state as a basis to outline succession of the origin of life stages. And I divided this succession into 4 stages. According to this approach, origin of life, origin of initial living systems occurred in the pulsating, during the pulsating migration of fluid to the surface in and 4 stages. First stage, non-living prebiotic microsystems, self-assembly with lipids and amino acids. Second stage, protocells, intermediate state between life and non-life, and the more or less equal contribution of energy and entropy, appearance on energy-giving process, respiration of redox reactions, and the local watering of membrane, and so on. Third stage.
5 minutes, Dr. Kompanichenko.
Okay, thank you. Third stage, primary living subcells, in which contribution from the thermodynamic point of view, the contribution of energy is prevalent, contribution of entropy already, and this is respiratory activity, the level of reactive oxygen species much increased, and appeared protein synthesizing apparatus and RNA, primary RNA templates. And the last stage, formation and growth phase cycle and genetic apparatus. And again, 1, 2, 3, 4 stages, and further evolution in the ocean. Key points. The transition to life is carried out through the intermediate state, which is maintained in oscillator mode, up and down the entropic barrier. Hypothesis. The succession of the origin of life process is recorded in the life cycle of a bacterial cell and repeats when it exits from anabiosis. And the primary population of living cells on Earth were formed in the process of pulsating migration of hydrothermal fluid to the surface, starting with the self-assembly of essentially lipid amino acid microsystems and ending with the formation of genetic apparatus of vegetative cells. And this is my acknowledgements to NoRCEL.
I would like to thank Sohan for his kind invitation. And let me start my talk by acknowledging the role of NoRCEL played in the research I am going to present you, because the second NoRCEL meeting in 2014 in Leeds gave me a great opportunity to conduct a conversation with Ram Krishnamurti about 3'5' cyclic GMP polymerization. And actually this conversation motivated me to move from theoretical work to experimental studies. And these experimental studies led me to think that actually crystallization could play an important role at the prebiotic production of the first biopolymers on our planet. Inorganic crystallization is a well-known process in nature. If you have a look at the rocks through a magnifier, you can immediately recognize that it consists of many tiny crystalline phases. On the other hand, organic crystallization is much less common, of course, but the inventory of modern minerals includes also a couple of organic members, and some of these organic members, some of these organic molecules which crystallize this way are as large as a nucleotide, for example. And from this, I thought that at the times when no biopolymers existed, organic crystallization actually could play an important role in the production of the first biopolymers. It could serve as a selection force at the accumulation of the building blocks of these biomolecules. People who are working in enzymatic catalysis are well acquainted with an effect which is named after the ancient Greek goddess Circe, which expresses the ability of enzymes to kinetically stabilize their substrates in high energy configurations, and this way to decrease the activation energy of chemical reactions. in the absence of enzymes in the prebiotic world, the crystalline order could play exactly the same role. So it could orient the molecules into favorable geometries, which enabled chemical reactions to proceed. In my talk, I would like to show you 3 examples for this prebiotic Cersei effect. The first one will be the polymerization of 3'5' cyclic guanosine monophosphate, which is a reaction that was for the first time reported from Ernesto de Mauro's laboratory. Nevertheless, we know that during the last 10 years, many of his original findings had to be revised. The second topic I would like to shortly mention is the polymerization of 2', 3' cyclic nucleotides. And finally, I will say a few words about our works related to the polymerization of the phosphoric acid salts of amino acids. Ring opening polymerization of 3', 5' cyclic guanosine monophosphate was reported to be a base catalyzed process. Nevertheless, our collaborative research with Dieter Braun's group has recently shown that the originally reported reaction is consistent only with an acid catalyzed mechanism because only the acid form material polymerizes. The first hints that crystallization could play an important role in the mechanism of this reaction comes from our computational studies. I have calculated the structure of the trigonal bipyramidal intermediate, which is formed when a nucleotide attacks a cyclic nucleotide. And I have found that in this structure, the distance between the glycosidic carbons is about 6.5 angstrom. And this is exactly twice as much as the optimum stacking distance in double-stranded DNA, from which I thought that we need a third nucleotide to stabilize the reactants into catalytically active geometries. And to my to my surprise, we have found exactly the same type of stacking interaction in the crystal structure of 3', 5' cyclic guanosine monophosphate. Then we decided to look at the morphology of the dry materials which were used for the polymerization reactions. And we have found that under acidic conditions, where the prevalent form of the material is the free acid form, this free acid form monomers form beautiful tabular crystals after drying. In contrast, under basic conditions, the sodium form material forms only an amorphous mass. Actually, it was not surprising for me that the free acid form monomers crystallized very well, because it is much more easy to self-assemble charge-neutral free acid form molecules than to self-assemble a sodium form material where you have to align nucleotide anions, their charge-compensating cations, plus the hydration sphere of these charge-compensating cations. And this is, of course, reflected also by the dramatic difference in the solubility of these two materials, because the sodium form material is about 100 times more soluble than the free acid form material. We have tested the polymerization reaction also on mineral surfaces. And interestingly, we have found that on those minerals where the nucleotides accumulated as an amorphous form, the polymerization yields were pretty low, and only very short oligomers formed. On the other hand, on those minerals where the material crystallized, we have observed formation of a large amount of pretty long oligomers, let's say. On the MOM 2021 conference, Steve Mojžić gave a wonderful talk about the relevance of amorphous rock glasses on the prebiotic earth, and he was very kind to give me some of his rock glass samples to test them in the 3', 5' cyclic GMP polymerization reaction. And actually from these studies, we have noticed a very simple relationship between the efficiency of the polymerization reaction and the metal oxide content of these amorphous rock glasses, because if the metal oxide content of the rock glass exceeded 50%, then the polymerization was significantly suppressed because the nucleotides basically dissolved the rock And this way they were converted to a salt form, and the salt form material didn't crystallize and didn't polymerize either. At this point, let me stop for a few words at the HPLC MS characterization of the polymerization products, which was done in cooperation with Dieter Braun's laboratory in Munich. And I am especially thankful for this work to Steve Unava, who is a very talented PhD student of Dieter. The analysis was done used a state-of-the-art. Sorry, why is it not moving? Stop moving, unfortunately. I don't know why. Okay. It used the analysis was done using a state-of-the-art instrumentation and the computer software, which enabled the identification of the chromatographic peaks, not only based on their retention times, but also based on the isotropic distributions measured in the mass spectrum for each chromatographic peak. But if you are still skeptical about the covalency of the oligomers in this polymerization process, let me show you our most recent measurements by Marditov spectrometry. In the multifragmentation spectrum of a mass spectrometric signal assigned to a PG-PG covalent dimer, we do see sorry for this technical difficulties. I have tested this before, and somehow, okay, we do see the main peak, which corresponds to the monophosphorylated guanosine, as well as a minor peak, which corresponds to the diphosphorylated guanosine. And the peak intensities of these two peaks are exactly the same as one would expect when fragmenting an authentic PG-PG covalent oligonucleotide standard. Okay. If you fragment the peak assigned to a PG-PG-PG covalent trimer, then besides the diphosphorylated GPG dimer, you can then you can also observe the phosphorylated GPG covalent dimer. And if you are still not satisfied, then let me show you our final proofs for the covalency of these oligomers, which comes from the hands of my colleague Aleš Kovařík from our institute. He managed to ligate a 24 nucleotide long DNA adaptor strength to the cGMP polymerization products. Another nice example for the importance of crystallization in the 3'5' cyclic guanosine monophosphate polymerization comes from our collaboration with Kate Slepokura from Wrocław University. Kate is a small molecular crystallographer who is very much interested in the transitions between the variously hydrated nucleotide crystals. She managed to convert the most frequently occurring trihydrate form to an anhydrous material, and interestingly, this anhydrous material polymerized maybe even better than the trihydrate form. But when rehydrating this anhydrous material, she obtained yet another trihydrate form, which was not the same as the original trihydrate, but it was very much similar. So we expected that it will polymerize too, but it didn't happen. We have repeated this experiment several times, always with the same results. And finally, we have recognized that this new trihydrate form is pretty unstable. So if we heat it, then it becomes quickly amorphous before it starts polymerizing. We have also studied phase separation of 3'5' cyclic cytidine and guanosine monophosphates from complex mixtures. When we made the Rivets cycling of an equimolar mixture of CCMP and cGMP, we have observed complete phase separation of crystalline CCMP and cGMP phases. And when dealing with 4-component mixtures, we have found that only the cGMP and CCMP phases separate as crystalline materials. The AU phases remain in the amorphous mass. And what is more interesting is that the we have observed also a spatial separation in the crystalline forms because the cGMP crystals accumulated around the edges of the sample, while the cCMP crystals grew in the internal, more hydrophilic parts. Now let me show you our most recent research related to the polymerization of crystalline sodium form 2' 3' cyclic CMP, where we have found that incubation in the vapor of ammonium carbonate induces formation of pretty long oligonucleotides from the crystalline material. Nevertheless, the polymerization yields are much lower than in case of an amorphous sample, even though in this case, the polymer lengths are shorter. We associate this with the different hydration degree of the dry materials. The amorphous material has a much higher water content. This is shown also by the Raman spectra of the crystalline and amorphous samples. As you can see in the OH vibrational region, the spectral bands of the amorphous sample are much broader than those of the crystalline sample, which shows a much higher hydration level. Actually, this higher hydration level of the material enables a better diffusion of the ammonium carbonate vapor into the amorphous material and higher reaction yields. On the other hand, the higher water content disrupts those key hydrogen bonds which keep the nucleotide monomers in reactive geometries in the crystals. And at the end of my talk, I would like to show you an old reaction in a new light. Phosphoric acid catalyzed polymerization of amino acids is known about for 50 years. Nevertheless, our recent research shows that using crystalline phosphoric acid salts of amino acids, the polymerization temperatures can be reduced by about 70 degrees. We have observed the best polymerization yields for the phosphoric acid salt of glycine. The conversion was around 10%, and the characterization was made by capillary electrophoresis mass spectrometric method in Oliver Traub's group at the Ludwig Maximilian University in Munich. And actually, for a solid phase reaction, 10% conversion is pretty good. And this is the consequence of two synergic effects acting in the crystal structure. On the one hand, in the crystal, the amino acid molecules are properly positioned for the peptide bond formation reaction. In addition, the phosphate chains, which are also present in this crystal structure, exert an enormous catalytic effect. This is the reason why in this polymerization reaction, we observe the formation of up to 24 mers. Just for comparison, let me show you what happens when you try to polymerize an amorphous mixture. Actually, it is not so easy to make an amorphous mixture when mixing together glycine with phosphoric acid, because the material starts to crystallize very quickly. We did this in two ways, with evaporation concentration at 120 degrees, as well as by acidification of a glycine sodium phosphate mixture. But the result was the same. After polymerization, we saw only heptamers, or in the best case, octamers. There is a discussion in the literature regarding the role of phosphorus acid in prebiotic chemistry, and this motivated us to consider also the phosphorus acid salt of glycine. Indeed, the crystalline material polymerized. Nevertheless, the polymers formed in this way are shorter, and more importantly, the reaction is impeded by diketopiperazine formation, because the crystal structure of glycinium phosphate enables not only the linear chain growth, but also the circularization of the dimeric products which form in the very first polymerization step. Let me note that in the crystal structure of the phosphoric acid salt, this reaction is not feasible, and this calls our attention on yet another advantage of crystallization that nature could utilize when producing the first biopolymers, Crystallization could help to overcome the circularization reactions, which is a well-known bottleneck of prebiotic polymerization processes. Let me note that we have also tried to polymerize other phosphoric acid salts. We consider the phosphoric acid salt of arginine as well as histidine. Nevertheless, in both cases, we obtained only pretty short oligomers, and the polymerization yields were also low. And there is a clear reason for this. If you look at the crystal structures, you can simply see that the monomers are not properly positioned for the peptide bond formation reaction. At the end of my talk, as conclusions, let me reiterate 4 major advantages of crystallization that nature could choose when creating the first biopolymers on our planet. Crystallization is a phase separation process, and as such, it could help to concentrate monomers from a highly diluted prebiotic pool. The crystalline state could preserve the monomers without degradation. For example, it could work against hydrolysis or depurination reactions. I have shown you a couple of examples that the crystalline state enables entropically favored reactivity in the absence of enzymes. This I call the prebiotic Cersei effect. And finally, the crystalline order could facilitate formation of longer biopolymers by preventing oligomer circularization reactions. At the end of my talk, let me thank to my co-workers on these projects. From the Institute of Biophysics, I would like to mention 2 colleagues, Aleš Kovařík and Roman Matyášek, who helped me with gel electrophoresis of oligonucleotides. From the Central European Institute of Technology, I would like to mention Ondrej Šedo and Zbigniew Zdráhal, who are always ready to measure for me Malditov spectra. We have an intense collaboration with Dieter Braun's group from the Ludwig Maximilian University in Munich on various polymerization-related problems. And from this collaboration, I would like to point out Sri Vunava, who measured basically all the HPLC-MS data, which I have shown you. Our collaboration network was recently extended with Kate Slepokura from Wrocław University, who gives the crystallographic expertise to this project. And finally, I would like to thank also to Ernesto Di Mauro, Giovanna Costanzo, and to you for your kind attention.
Let me say something about the outline of my talk. Firstly, I would like to talk about origin of water on the Earth, and a little bit about the history of water. And secondly, I want to talk about stellar wind contribution to origin of water in space. And then I want to show some laboratory experiments which are related to origin of water, on which we can somehow create on the surface of oxygen-containing minerals. And finally, I would summarize my talk related to the water and discuss the origin of water on our planet. Here, I want to show famous picture, which is showing history of our universe, and from the Big Bang until now. And according to water and origin of water in the universe, we know that the standard cosmology, hydrogen was formed immediately after Big Bang, and which is around 3.8 billion years ago. And after just 1 millisecond, protons and neutrons emerged from the cooling quark gluons plasma, and the next atoms, like oxygen, was not created, and hydrogen to create the water has to wait about half of a billion years after first stars has formed, and because oxygen was the product of fuse on big stars. This slide shows one of the oldest spectra which was measured, and you see the ALMA spectrum from galaxy SPT 0346, and which is you can see the pure rotation lines included, also ionic water lines together with carbon dioxide lines. So this spectrum has a red shift which is about 5.66, and from this we can calculate the age of the spectrum, which is about 1 billionth of years after Big Bang period. So let me talk a little bit about the origin of water, and where actually origin of water on our planet, and actually there are two views or two theories about origin of water. One theory is that water was created at the beginning of formation of our planet in the solar system, and it survives until now. And the second theory is talking about event when Earth collapsed with some asteroid, and Moon was evolved, and this theory somehow tells that some water was delivered through volatiles by asteroids and comets during the initial stages of our planetary differentiation. So nowadays there are some data available, and from precise measurements in space, and one thing which can help about our understanding of origin of water is somehow the measurement of D to H ratio, and which is a factor which is somehow related to origin of water from the universe. We have if you look here and we measure this D to H ratio on our planet, we get the value which is something about 1.5 times 10 to minus 4. And this ratio is so-called ocean standard. And if we look outside of our planet, for example, to Jupiter's and our planets, we can find that there is a deficit of deuterium, and it's less concentration of deuterium. On the other side, if we look at some objects like a comet, like Churyumov-Gerasimenko, we can get a ratio which is much higher. We can get a much higher concentration of deuterium in this kind of material. And secondly, or thirdly, if we look at carbon-containing chondrites, and so we can find almost the same ratio to this ocean ratio on our planet. So this is just, or maybe this is better picture. If we look at the concentration D2H ratio on comets, you see this high concentration. Here, this blue line is showing our concentration on the Earth. And if you look at the planets like Jupiter, Saturn, Uranus, and Neptune, we can see a higher concentration of hydrogen opposite. And very close to concentration, ocean concentration, D2H is coming from asteroids and meteorites. Explanation for is very easy because this is main role is playing this kind of equation that this equation is showing it's showing that this equilibrium is going on both sides, and if material is coming cold cold interstellar cores at temperatures which are less than 50 kelvins, then they are much more deuterium on these surfaces. And opposite, if this material on asteroids or meteorites are coming close to some hotter regions, then this reaction is somehow shifted to hydrogen, and this material is containing much more hydrogens in comparison to deuterium. So let's talk about our laboratory experiments, and this is just a picture. This is showing what we are going to talk about. So you have stars and or some we can say this is some star, and we can see stellar radiation. We know that stellar radiation mostly contains hydrogen ions, and if we bombard the body of asteroids, and these asteroids contain some minerals, oxygen containing minerals, then water can be created on surface of this body. And then by impact of these bodies on some surface of planets, this water can be distributed to the planet. Actually, we did in laboratory two different experiments. One experiment is so-called thermal programmable desorption with mass detection. And in this case, we have a sample, and this sample was bombarded with ion gun with some ions. Here we use either hydrogen or deuterium ions, and here some materials which has been bombarded. So, for example, the titanium oxide with oxygen 18 was bombarded with hydrogen, and we look at hydrogen water 18 product. And then opposite, we take titanium oxide with oxygen 16, and we bombard the surface with a deuterium atom. And at this case, we look at D2O molecule, and so on, and also the others materials. If we look at the profile, which is interesting, that here, for example, this is the example of titanium oxide, which was bombarded with deuterium atom, and you see the production of D2O. And then you see that we change the temperature of the sample after the bombardment, and then we see that the water is dissolved from the sample at temperature around 400 Ks and 400 kelvins, and we detected in this case D2O molecule. So here another example. This is iron oxide, and similarly you see the maximum peak at around this 400 kelvins. And just by summarization of this experiment, it's interesting that if we summarize that, we see that there are quite strong forces which keep the water on the surface, because everything was done in a vacuum chamber at pressure which is below 10⁻⁸ or 10⁻⁹ torrs, and so this is very high vacuum, so which somehow demonstrate that water can be bonded strongly on material, even in the universe, in conditions of universe. And then you see this quite high temperature, so it's even at 400 K starts to dissolve from the surface. So this is something showing that if such a body which can go close to some star, that can, in this case, water can be created on the surface, and then transported through the universe for a long period of time, and transported completely to some completely different place, and even through this transportation, water can be permanently produced. So this is what was related to this TPD experiment. And secondly, we were interested about quantification of the water, amount of water which is bonded to the surface of this oxygen-containing minerals. For this case, we use high-resolution Fourier transform spectroscopy, infrared spectroscopy, and we built a special experiment, which is maybe better to shown on this figure. We have an oven here. We have a quartz finger here. Inside, it's this oxygen-containing material. We use a microwave discharge, so we can discharge it hydrogen or deuterium here, and we can irradiate the sample here inside. And then finally, by heating of this finger, we can somehow decompose the water from the surface, and we can measure the absorption spectra directly from this sample. And this is just high-resolution spectrum showing that we can create in this hydrogen discharge, microwave discharge, this hydrogen Rydberg transition or atomic lines of hydrogen. Here, this is a table which summarizes all samples which has been studied. We took different minerals, and including 2 meteorites, and all of them were irradiated either with hydrogen or deuterium ions, but mostly with deuterium to create D2O, heavy water. So this is a picture. This is showing this microwave FTR experiment, and you can see the different types of synthetic materials which were bombarded by the deuterium atoms, which were generated in microwave plasma. And here you see the standard, you see the heavy water spectrum, high-resolution heavy water, which is in the infrared spectral range. This is this OH or OD band of heavy water. And then you see the spectra, which has been collected from this kind of a material, montmorillonite, kaolinite, and augite. And this is a just comparison with the standard D2O spectra. We did all of them, we measured all of them. You can see that there are different concentration of water by this bombardment. So these materials, they behave a little bit differently. And finally, we were able to quantify the amount of from the spectra, from intensity of the spectra by fitting of the spectra, all these absorption lines. We were able to estimate amount of water bonded on surface of these oxygen-containing minerals. We want to compare, the subject of our idea is just to somehow to support the theory of transport of water from the space. And we know that in period 3.8 billion years ago, it happened, so-called late heavy bombardment period. So our planet was bombarded with heavy bodies, asteroids, and so on. We can see such thing if you look at moon. So you see plenty of craters here on the moon, and by mapping of these craters and size and so on, we can somehow estimate amount of bodies which has been transported on surface of moon during this heavy bombardment period 3.8 billion years ago. And if we extrapolate this amount of mass of bodies to our planet, we can get something, some calculation which gives us some information from astronomical point of view about amount of material which was transported during this late heavy bombardment period. And we come to such a number, which is something about 1 × 10 to 20 kilograms of accreted materials during this late heavy bombardment period. So please memorize this number here. And if we look at this table, so you can see the table which is summarizing the materials which was studied. So for each material, we got so-called adsorption capacity from our measurement, laboratory measurement with application of this high resolution spectroscopy. And here we can, from this water absorption capacity of sample, which has been treated by microwave discharge, we can somehow calculate it or get the amount of accreted material which it's required to bring in one equivalent of amount of ocean water on the air. So in other words, I can simplify this thing that how much this material do we need to fill all oceans on our planet. So for each kind of a material. So you see that according to this iron oxide, we need something about 9 × 10 to 19 kilograms of material. So we are, for each materials, we come to numbers which are in order of 10 to 20 kilograms of this material. So this is, and if you memorize this number, which was somehow calculated by astronomers, you can see this is a very good agreement of our estimation in comparison with astronomical number. So I'm coming to the conclusion, and let me conclude my talk. So we studied about 14 different samples, including 2 meteorite samples, and we are showing that there is a reaction with a solar or stellar radiation on the surface, and we can really create water on the surface. And in our case, the capacity.
5 minutes, Dr. Sivis.
Okay, thank you very much. It's about in this kind of weight. It's from 0.1 to 0.7%. But we have to take in account that we have a limited time for irradiation in the laboratory, so we are limited by time. So we irradiated with maybe a couple of hours. We irradiated the sample. But if you compare to space experiment or space condition, then you have to take in account that some asteroids they can fly in the universe billions of years around the stars, and still the water can be permanently created on the surface of these bodies. So this is one thing. And second, the absorbed water from this TPD experiments remains on the surface at very low pressures, about such low pressure. Still water is very somehow bonded to the surface, and it can be evaporated at temperature around 600 kelvin, so slightly quite high temperature. So even at high temperature, water survives on the surface. So these 2 things, they suggest that about the possible transfer over long distances and over long time scales in the universe. The next point is that our measurement has a general character and demonstrate that any interactions of oxygen-containing minerals with stellar radiation comes to or leads to generation of water, which can be absorbed on the surface of minerals. And the final thing is that if the surface of material were fully saturated with absorbed water, and from our calculation suggests that during this late heavy bombardment period, amount of water equivalent to more than 1 of Earth ocean could have been transported on surface of asteroid. So there is a possibility that easily all ocean can be completely filled with water during this late heavy bombardment period. So thank you for attention. And recently our paper was published in Astrophysical Journal, and here is address of this paper, which can be read in general open access.
Happy to be here to describe some of the studies we have been performing on these fascinating rocks. Today we have been hearing really interesting talks describing these meteorites that are accretionary. They are part of the first materials that were surrounding our sun. In particular, in this talk, I will be just describing some really amazing properties that these rocks have. In particular, in this diagram, in this first slide, we have on the left side the different organic compounds that you can obtain just by performing the experiments with it, just removing all carbonaceous, all organic materials from these carbonaceous chondrites, and just making experiments with water and formaldehyde. In only 24 hours, we were able to recover all these acids, nucleobases, and analogs, amino acids, and miscellaneous material that makes us really enthusiastic about these catalytic properties of carbonaceous chondrites. In particular, I will explain how it's possible that these meteorites are able to catalyze all these organic compounds from zero. The reason for that is that in the components, when we are going to the nanoscale, as we can see in the Murchison meteorite, we find that these materials were not affected by many processes. They are practically untouched, and they host some properties that were having at the very beginning of the solar system, just probably providing this kind of link with the stellar environment. Just let me go. I don't know why it's not going. Let me see. Okay.
To the left, to the left.
Yes. Okay. I think it was so white I was unable to find the right button. Okay, so a basic outline of the talk will be, first of all, describing the evolution of organics from pre-stellar clouds until protoplanetary disk, describing the formation of the solar system, the first stages, how important was the stellar contribution, the stars that were surrounding the sun at the moment of formation. Then I will give some clues about the condensation of the materials forming these meteorites as the origin of the rock-forming materials of planetesimals and cometesimals. At that really early stage, we have a competition between accretion versus disruption of the building blocks of planets at the end, hundreds of millions of years after these processes. And of course, the temperature gradients, the irradiation environment had a very significant implication on the condensation of the minerals forming these meteorites. Then I will describe some of the chondrite groups as samples of the protoplanetary disk. And from there, I will go describing the hydrated groups of carbonaceous chondrites that are very relevant in the sense we have materials that are accretionary, but containing also hydrous phases, and particularly organics. And then when some of the experiments to decipher the catalytic properties of chondrites. In a sense, these are unique samples from the outer protoplanetary disk. And recently, these fascinating missions, Hayabusa 2 and OSIRIS-REx, were able to sample return some of these asteroids that, in fact, are opening a new door to future amazing discoveries and correlation with carbonaceous chondrites we have in our laboratories. And in general, what are the implications for the origin of life on Earth? and elsewhere. So we can see in here, we have, since the very starting moment of the prestellar of the molecular cloud, in which some stars are formed, and then these stars nearby are ionizing the nebula, are producing just the collapse of other small parts of these molecular clouds that can be thousands to millions of solar masses. And finally, locally, we have the formation of these protostellar clouds that finally become protoplanetary disks. In these protoplanetary disks, we have the condensation of planetesimals, the condensation of minerals that finally are forming planetesimals. Everything starts from quite simple molecules because this is an ionized medium. In the protostellar phase, we have more complex molecules. And in the protoplanetary disk, we have simple and complex molecules depending on the region of the disk. If this is most, ionized by the really extreme environment surrounding the sun, we will have more simple. And we have some regions that are hidden from all this heat and all this radiation, not only emitted from the sun, from the central star, but also from nearby stars that can be really close by the star. And well, during the first millions of years of the solar system, when condensation occurs because of the fast cooling of the protoplanetary disk environment as consequence of after the birth of the sun, we have the sun emits a lot of radiation, several orders of magnitude in each one of the windows, electromagnetic windows. So we have really a cleanup of the gas that makes the temperature decrease very, very quickly and condense tiny micron to nanometric minerals, forming, depending if we are really close by the sun, are dominated by metal grains, sulfides, silicates, because are rocky materials. But if we are going to the outer part of the protoplanetary disk, we have some self-shielding by the own materials that are mostly in the equatorial plane of the disk. And then in there, we have the condensation of organics and ices that participate in the formation of comets or cometesimals. In these bodies, also depending of further reactions were happening, mostly because some of the materials were hosting radionuclides, and these radionuclides provide heat, make higher molecular complexity. This is something that was happening in these environments. So we have different pathways towards complexity, towards organic complexity. And to understand them, then we have to really understand the different stages. There is this excellent review by Caselli and Ceccarelli from this synthesis from that review, saying that the evolution of the molecular complexity goes hand in hand with the physical phases involved in the formation of solar-type planetary systems. So let's talk a little bit about planetesimal formation. The primordial components, we have the sun and the protoplanetary disk from the molecular cloud. We have clear evidence that in the solar system formation was happening nearby, close by a stellar association, or even could be a stellar cluster. And the reason for that is that in these primeval meteorites, we are not having not only having these condensates that are formed surrounding the sun, but we have other tiny grains that are called stellar grains or presolar grains that are hosting anomalies, isotopic anomalies that we can directly relate the formation of these grains in the envelopes of these nearby stars. Envelopes of different types of stars that we know very well thanks to astrophysics, in which we can really know the origin, not only the origin of these stellar grains, but also the origin of short-lived nuclides, these radioisotopes that were the key to hit internally asteroids and, of course, planetary embryos, with the relevance of this internal heat to our understanding of differentiation of terrestrial planets. So, just very summarizing this idea, the main sources of that are circumstellar cells of red giants, asymptotic giant branches, stars, novae and supernovae ejecta. And of course, the outer disk, the outer region of the protoplanetary disk also incorporates organics and ices. So in here we have just a brief compilation. This is from another review, really very relevant, from Earth's Thinner, in which we have the different short-lived nuclides. These are the parent and the daughter. So we have each one of these short-lived nuclides that decay in a half-life of millions of years, typically, or less, and that has an influence, particularly 26 aluminum and 60 iron, in because they were more abundant than the other short-lived nuclides, in hitting these planetesimals. So in the protoplanetary disk, at early times, the gas drag produced migration of bodies. We have the condensation of all these minerals falling into the equatorial plane. And then we still have some significant amount of gas and, of course, tiny grains of dust that are participating in, making that the materials that are formed are falling against the sun. So little by little, they are just also affected by heat, and they are partially volatilized, and of course, during these first stages, first millions of years in the evolution of the protoplanetary disk, were the key to understand the formation and the characteristics of undifferentiated asteroids and comets. In particular, we know that all this process was blowing off sodium, potassium, sulfur to produce volatile enriched cometesimals. And I will present very clear evidence we have recently produced, studied and we have published in Nature Astronomy on all these processes that were happening at the very beginning. So, and thanks to all these new interferometers like ALMA and SCA, we can infer the chemical gradients, the different structures, the different rings that are surrounding another stars. This is like coming back in time 4,567 millions of years to really see how our own solar system was made at that time. No planets, no asteroids, no comets so far, just rings of materials surrounding the star. In these moments, some of these tiny minerals started to suffer heat by collisions, by friction in the shock waves that were produced in the protoplanetary disk, and of course, strong flares from this really energetic sun. The early sun was known as one of these stars that we call T Tauri stars. And all these participate in processing these materials and producing tiny drops, tiny contours, millimeter to centimeter size, that are formed mostly by olivine, primordially fosterite. But as consequence of heat, this primordial fosterite was changed by the presence of iron oxide in the environment, in the gas, was little bit transformed into phyalite. Then we have pyroxene, enstatite, plagioclase, troilite, etc. This made these contours, these igneous texture particles that are composing most of these chondrites. That's the reason for the name of chondrites, because they are formed by contours. In between the chondrite groups, we know different petrologic types, from a type 3 that corresponds to the middle of this diagram by Elizabeth Dotto et al. This is a very basic, simplifying everything. We have materials that have suffered metamorphism, like marble in the earth by subduction, but just by overpressure of the materials sink into the asteroid. So higher temperature, higher pressure, higher thermal metamorphism. But the groups that we are interested in that have not suffered thermal metamorphism are these carbonaceous chondrites. And particularly, they have suffered as very significant action of water. And this is what is here explained as aqueous alteration. From the type 3, we go to type 2 and type 1. That is the stream aqueously altered material. So we have carbonaceous chondrites, rocks from these water-rich bodies that can contain even more than 10% in mass of water and about 4% in mass of carbon, most as insoluble organic matter forming the matrix of these meteorites. And these experience aqueous alteration at different degrees. I will just give some examples very quickly. Of course, we have all this new evidence from Ryugu, obtained by Hayabusa 2 JAXA mission, that achieve sampling at 2 different locations. And then we also have, well, these materials are similar to heated CI chondrites. And of course, Bennu, that has been sampled by OSIRIS-REx mission, that is also the smallest orbited celestial body so far. It's also similar to CI CM chondrites, and is extensively aqueously altered, hydrated minerals, even on the surface of the asteroid. And this is what we were studying. I was at the Institute of Geophysics and Planetary Physics at UCLA for more than 2 years, working on carbonaceous chondrites, and we were describing the different stages of alteration of these chondrites. In particular, we found that they were probably altered due to their burial depth, and at the same time, the samples were brecciated, fragmented and compacted by further impacts since their formation. Then, just returning to
5 minutes,
Catalonia, thank you. We found the interest in just studying this Murchison meteorite. We can see here different phyllosilicates that are explained in here in these boxes that are in contact with really active minerals, like, for example, we have here iron and sulfur, so we have thoylite, pyrrhotite sulfides, calcium carbonates as well. And of course, this is just in the same matrix. We are having in here 200 nanometers, so this is tiny sample in the matrix of this meteorite. This is evidence probably for wet accretion of these phyllosilicates into Murchison meteorite. Here's how we use the diffraction patterns to understand and characterize the different phyllosilicates. In CRs, we have also observed extensive aqueous alteration. We can see, look at these clusters of magnetite. In here, we have pyrrhotite that has been altered completely by water, by the action of water, at the very beginning of the accretion of this asteroid. And in here, we have all this, all this evidence for hydrothermal activity in the parent bodies of carbonaceous chondrites by different authors. Just here you will find some examples. In particular, Keynes and Smith were proposing that the phyllosilicates could be synthesizing light. Well, what we know, we have obtained a series of experiments in which we demonstrate that the Fischer-Tropsch process, a collection of chemical reactions that convert a mixture of carbon monoxide and hydrogen, molecular hydrogen, or water gas into liquid hydrocarbons, and they occur on the presence of metal catalysts. And this is what we can find in carbonaceous chondrites. So what we were planning to do? Well, we studied the catalytic effect under the action of water of 6 carbonaceous chondrites. These 6 carbonaceous chondrites are listed in here. We were just studying, thanks to that our center has been recognized as International Repository of NASA Antarctic Carbonaceous Chondrites, and we were able to, grind these meteorites and just subject these powders into hot water and formamide. This is a synthesis of the experimental conditions. So what we can found is that even removing previously all organic compounds, you can reproduce in only 24 hours if you put this formamide with water, hot water, and these tiny grains of carbonaceous chondrites. So they are hosting catalytic properties unknown in any other type of rocks, not in Earth, not in Mars, not in Vesta. So these really primordial minerals incorporated to these chondrites, probably as they were raining as thousands of tons at the beginning of the time, on the Earth, on Europa, on Titan, on Mars, probably they had the chance, if they found a really warm environment with liquid water, with amides, with presence of nitrogen, just the right conditions to probably promote chemical complexity. How these participate in the origin of life? At least they produce this soup that really can go and increase little by little our understanding. And well, in this meteorite, as I was presenting, we discovered this tiny clast. It's a xenolith, a fragment of another body, probably a comet or a trans-Neptunian object, that when incorporated into this CR chondrite that is called Lapath 02342, and we published with the team of Larry Nidler the analysis of these materials. It's it was very amazing that these clasts have a composition unknown so far, in which we can find a lot of sodium, sulfur, and other really moderately volatile elements that probably were at work making really unusual chemistry in the other part of the disk from which this material was condensed. This is the long pathway of this ultracarbonaceous class, this xenolith, in which incorporate into the other part of the protoplanetary disk, just went with an ice mantle forming a comet, and when the comet disrupted, was finally incorporated to the CR parent asteroid until the meteorite delivery to Earth. So in conclusion, chondrites are fragile and porous meteorites that were able to retain the chemistry of the primeval disk environment in which they form. So the different chondritic components host clues of different formation regions as stages. These meteorites accreted organics, water, and catalytic minerals. And hydrothermal asteroid activity is confirmed from the nature of the soluble organic compounds and their non-terrestrial signatures. In particular, carbonaceous chondrite minerals participated under water presence in the synthesis and interconversion of organic species of prebiotic origin. The matrix of these carbonaceous chondrites was the source of prebiotic compounds, and that synthesis could have been predated, at least partially, the formation of the Earth. That's very important. And the results of these catalytic experiments indicate that carbonaceous chondrites catalyze the synthesis of natural nucleobases, carboxylic acids, and amino acids from mixtures of formamide and water at about 140 degrees Celsius. The two general scenarios analyzed, thermal water and seawater, we tested the experiments with different types of water, were all favorable to different degrees. And these carbonaceous chondrite minerals could have been widely distributed in the solar system because these materials reign over the planetary embryos. So life perhaps had several potentially favorable locations to develop besides the Earth. Could be Europa, Mars, Titan, etc. So thank you very much for your attention. We'll say that the space exploration as the key to understand life appearance.
Okay.
Thank you.
So here's the preview of the content, what I'll be discussing throughout this session. And since this is a conference themed on reactive oxygen species, I just want to note that this study did not involve direct ROS experiments, but it potentially would provide a sort of critical model for understanding how organic matter in extraterrestrial environments, often exposed to ROS generating conditions like cosmic radiation, might evolve. So by synthesizing and characterizing IOM, this work sort of lays the foundation for future studies on ROS interactions with organic molecules relevant to astrobiology and prebiotic chemistry. So to start with, what is IOM? I think by now, if you guys have been following the previous sessions, we probably should have already an idea on what IOM is, thanks to the previous speakers. But let me also shed a little bit of light. So IOM, or insoluble organic matter, is a class of complex, high molecular weight organic compounds that are chemically resistant to dissolution, so they're difficult to be dissolved. And what makes IOM particularly interesting is its unique stable structure, composed of both aromatic and aliphatic components. And this stability has allowed IOM to survive in harsh conditions where it provides insights into the organic chemistry of the early solar system. And so where is IOM found? So IOM is commonly found in carbonaceous chondrites. We've also heard this throughout the sessions earlier. It's a type of meteorite rich in organic material. And besides meteorites, we can also find IOM in planetary soil such as Mars, in cometary dust, and even in interstellar dust grains. And the presence of IOM across various solar system bodies make IOM a crucial component for studying solar system evolution and organic matter distribution. So why is IOM important again? IOM is very much central to astrobiology and planetary science for several reasons. The first one being it offers insights into prebiotic chemistry, providing clues about the types of organic reactions that might have led to life as we know it today. And secondly, IOM might also have preserved ancient organic molecules, giving us a window into conditions of the early solar system. And thirdly, by studying IOM, it can also help us interpret spectroscopic data collected from various space missions. And this could further improve our understanding of extraterrestrial organic matter. However, studying natural IOM directly can be challenging due to its heterogeneity and limited availability. Unless if you're cool like Professor Joseph earlier, like they have access to direct combination rights, so this can be a challenge. So therefore, our approach here is to sort of synthesize IOM analogs in the lab that mimic the key structural and chemical characterizations or characteristics of meteoritic IOM. So therefore, at the lab, the main aim of this research has been to synthesize a collection of IOM samples using various starting compounds, namely paraformaldehyde and glycoaldehyde and nitrogen, along with other elements like sulfur and phosphorus. So far, I've been working with nitrogen only at this point. And upon synthesizing, we aim to analyze the samples' spectroscopic features to compare them with natural IOM found in meteorites. And this comparison could highlight similarities and differences, pointing to the different unique properties that could arise from cosmic synthesis processes versus lab-based conditions. So ultimately, our goal is to sort of build a database, a mini database of synthetic IOM samples that can eventually aid or help in analyzing spectroscopic data from planetary soil and meteorite samples. So the sort of hypothesis that we have at the moment is like, we hypothesize that by adjusting the precursor molecules or the precursor ratios of the molecules and experimental conditions, we can sort of create synthetic IOM analogs that reflect the structural and functional characteristics of natural IOM. And specifically, we expect these analogs to replicate some of the spectroscopic features of IOM found in meteorites. And IOM could have also served as a primordial multifunctional organic entity or prime, which could provide a universal basis for the key requisites of life in terms of compartmentalization, metabolism, and replication. So what have we done? So this is a broad overview of the processes or the steps we have taken. So we began by preparing the starting solution, followed by thermal treatment, separation and washing, freeze-drying, and finally desiccation. And after the whole synthesis of the IOM samples, they were then put through a series of spectroscopic analysis to evaluate their chemical structure. So the first step, what we did was to prepare the starting solution. We dissolved paraformaldehyde and glycoaldehyde in distilled water, and we adjusted the pH to be in the range between 11.5 to 11.7 using calcium hydroxide. And this table here, you guys can see, it shows the different ratios of the precursors used for each sample, labeled from E1 through E6. So we varied the glycoaldehyde amount as well as ammonia to see how these variables would then influence the resulting structure. And we've used like paraformaldehyde and glycoaldehyde as they are very simple organic molecules that can mimic the early building blocks of life. And next, the solution was then subjected to thermal treatment. We heated it to 180 degrees C for about 2 hours using a microwave mineralizer. And this high temperature initiates polymerization, leading to the formation of complex organic structures within the solution. And this step is critical for driving the condensation reactions necessary to create the IOM analogs or the IOM samples. And after the thermal treatment, we centrifuged the solution at 8,000 RPM to separate the solid insoluble organic matter, the IOM, from the soluble organic matter, or SOM. We then washed the IOM solid twice with hydrochloric acid and twice with distilled water to remove residual soluble material. And this is the IOM extracted from the soluble organic matter. It looks like a seaweed structure here. And this washing step here, it ensures that our final product is a clean, isolated form of IOM. And then the IOM solids were then freeze dried, and we obtain some sort of powdered form of solids in this form, as can be seen in this figure. And these solids were then further dried with sodium hydroxide to eliminate any remaining moisture. And these drying steps, they're essential to stabilize the IOM structure, making it suitable for spectroscopic analysis. So what we end up with is a purified, stable form of synthetic IOM. Now that we have obtained these IOM solids, what do we do next? How do we know what's inside the synthesized IOM solids? So this is where the spectroscopy techniques come in. So we can say spectroscopy is like a molecular detective. So by shining different types of light, we can peek into the structure of our IOM at a quantum level. So from NMR to gamma ray, each method would reveal a piece of the puzzle. And so once again, the overall methodology, and now moving to the spectroscopic analysis, specifically to analyze the complex structure and composition of the IOM samples that we've obtained. We applied this following our spectroscopic techniques for the moment. And each of these techniques, they operate in a different region of the electromagnetic spectrum, allowing us to examine the various aspects of IOM at both molecular and atomic levels. So firstly, we started with the ATR, FTR, which stands for the Attenuated Total Reflectance Fourier Transform Infrared Spectroscopy. It's a bit mouthful, but yeah. So this technique uses the IR region of the spectrum to detect molecular vibrations. And by identifying the absorption peaks, ATR, FTR reveals the specific functional groups of the samples, such as aromatic rings and hydroxyl groups. And this is crucial because it gives us insight into the types of chemical bonds and structure present in IOM. The next technique is the nuclear magnetic resonance. It works in the radio wave part of the spectrum. So this technique specifically targets the carbon-13 nuclei in solid samples. So it would provide us information in terms of the carbon environments of the sample, allowing us to examine the structural details like aromaticity and branching. Also, these details are essential for understanding the complex carbon network in IOM, which is highly relevant to its overall chemical behavior and origins. And then we've also used UV-vis spectroscopy, and it clearly operates in the ultraviolet to visible spectrum range. So this technique, it measures the electronic transitions within molecules. So for IOM, UV-vis helps us observe electronic structures by identifying the bonding and anti-bonding transitions, which are common in aromatic compounds. This technique also lets us compare different precursor ratios, showing how they would affect the electronic properties. And then we have the EPR, the electron paramagnetic resonance, which uses the microwave region to detect unpaired electrons in the paramagnetic species. So this is particularly valuable in our study because it measures radical concentrations. So in short, all these different techniques gives us unique insights into IOM by focusing on different spectral regions and molecular characteristics. So together, they would help to create a detailed picture of IOM's chemical structure, reactivity, and potential similarities to extraterrestrial organic matter. And in terms of data, let's look at some of the data that we have obtained so far. So these are from the preliminary results. So the FTR spectra, here you guys can see, it showed strong peaks corresponding to aromatic rings. The first curve, it's the IOM with nitrogen, and the one below without nitrogen, but it still shows the peaks corresponding to aromatic rings, confirming the presence of these functional groups across the different initial samples. While on the right, it's the NMR spectra. It provided us with insights into the aromatic and branched carbon structures in the initial IOM samples, also indicating a complex carbon framework that's consistent with natural IOM's aromaticity. And then moving on, in terms of the UV-vis spectroscopic results, we have, the UV-vis data reveals distinct electronic transitions across different samples. So the one on the left is samples E1 to E3, on the right, E4 to E6. They also demonstrate strong absorbance at lower wavelengths, around 270 to 300 nanometers. So they suggest high electronic transitions, typically associated again with the bonding anti-bonding transitions in aromatic compounds. And here, E1, the blue one, if it's visible, it's the topmost curve. E5 is the bottommost, and E6 is the center. So these, the differences between these samples, especially in terms of intensity and slope of the absorbance, decay, they reflect the sort of variations in precursor composition, which influence the electronic structure. And then we move on to the spectroscopic results for EPR. Okay, sorry, I do not have the high resolution of this picture. I was not able to get them on time. So this is what we have. So the EPR spectra here, they indicate high radical concentrations across all samples, with notable differences between the carbon-bearing samples E1 to E6 and nitrogen-containing samples E4 to E6. And nitrogen-containing samples on average, and look here at the table down here, they have lower radical spin concentrations, but increased delocalization over here. And they are attributed to nitrogen's stabilizing effect on radical structures. So nitrogen addition reduces the concentration of unpaired electrons, but spreads them more uniformly, suggesting enhanced radical stability through nitrogen's influence on the ion structure. So these are the sort of spectroscopic results that we've obtained so far with all this different spectroscopy. Moving forward, we still have the Maldi mass spectrometry and Raman spectroscopy to be done. Also for the second batch of the starting compounds with sulfur, and the third batch with phosphorus. So yeah, and to sort of like wrap up, like so far, it's promising. I mean, that this research can sort of like provide valuable insights into the structural and chemical properties of synthetic IOM, and has demonstrated so far the effectiveness of using multiple spectroscopic techniques to create sort of a detailed profile of these analogs. And ultimately, once again, this work aims to, we sort of like aim to have like a mini database of these different synthetic IOM analogs, and then to be able to compare them against the actual meteoric data coming from different missions. And with that, I thank you. Also, shout out to the Vaalbara Chemistry Lab, and also Lorenzo, who's there on his graduation. He's a master's student right now. I've been working together with him also for the first half of this project. So, any questions? I'm ready.